Recording in progress. All right, good morning, everybody. We're going to go ahead and get started. My name is Allie Dunn. I'm a senior environmental scientist with the State Water Board Surface Water Ambient Monitoring Program, or SWAMP. And yeah, it's good to see you all. I see a lot of familiar faces, some new faces too, and it's just good to be back in person doing this thing again. Let's see. So I'm going to kick off the welcome and then hand off to John on behalf of Society for Freshwater Sciences, but I'm kind of representing the California Aquatic Bioassessment Workgroup piece or half of this meeting. So just real quick, the bioassessment program, I think many of you are familiar with the program and the work that we do. But for those that are not, I thought it'd be helpful just to give a little overview. So the gist of it is that we use biological data to help understand stream health and then to help inform management decisions for the water boards and other folks. The program is well established. We've been around for over 20 years and have been monitoring water bodies statewide, just like I said, to kind of get a sense of the biological conditions statewide. And then because I work the water boards, this is also to help support our mission, which in part is to protect and restore the ecological integrity of our watersheds and water bodies. So while the program does lots of things, I thought I'd just kind of touch on our core monitoring work that we do. There's 3 areas that we're focusing on. The reference condition monitoring program, which we go out to sites that are minimally impacted by humans, and that data is used to kind of serve as the benchmark of biological conditions statewide. We're putting a lot of energy, and you'll hear from Nathan Mack at Fish and Wildlife and Chad Laughlin at the San Diego Regional Water Board into our non-perennial streams assessment. So going out to the systems that are intermittent and trying to collect some data there for those guys. And then finally, the perennial streams assessment. This is a program that we're backing off of just a tiny so we can focus on the non-perennial stuff. But this data is used to get a sense of status and trends of biological conditions statewide. And last thing I wanted to say here is this program takes a lot of work, and it's a close partnership between the water boards, folks at Fish and Wildlife and the Aquatic Bioassessment Laboratory, CSU Chico, Southern California Coastal Water Research Project, and Moss Landing Marine Labs. And you'll be hearing from some of those guys later today as well. Okay, so for today, I'm not going to go through all the details here, but you're welcome to take out your phone and scan the QR code, which is also here on the inside of your agenda if you have that printed version. I'll be kind of handling this first chunk of the meeting. We'll have some programmatic updates after John does his welcome piece. And then we have a keynote by Caitlin Boise. And then we'll get into a talk on bioassessment and intermittent streams. Let's see. John will talk about at the end of the day, there's going to be a business meeting for SFS, as well as a social mixer. And then on the second day, I think Elena let some folks know, but we had some changes in the agenda. So if you go to that electronic version, things should be up to date there. If you have an old school version and you want to mark it up, you're welcome to do so. But we won't be starting until 9 a.m. tomorrow. And then one of the talks in the afternoon, or not afternoon, the second session presentations, he's with USGS. So federal government shutdown, he can't make it. So those talks will be bumped up 20 minutes and we'll break for lunch at 12, not 12:20. So disregard this slide. And then Ryan Peak will be hosting an art workshop in the Klamath room tomorrow. Okay, some housekeeping things. If you need to use the restroom, go out these double doors here to your left and you'll see signs. There's also some water refill stations out there for folks that brought their own water bottle. You can refill there. And then John and SFS have provided snacks, and those will be out at the front table. I don't think they're set up quite yet, right? Okay. And there should be coffee and all that good stuff. And then finally, during Q&A, we are hosting this through Zoom as well. So use a mic. There will be folks running mics. Make sure you talk into it. And last couple of slides, I just wanted to thank the planning team. I'm on there, so I'm not thanking myself. That was just pulled over. But yeah, huge thank you to the planning team. It was truly a team effort, and through the leadership of Elena, who really paid attention to every small detail. It's a lot that goes into this, and all those little things behind the scenes, she just really did an amazing job. So thanks to Elena. And to all of you all. So this wouldn't happen without all the volunteers and speakers and participants. So just sincere gratitude to you all for making this year's CABW happen. And John, over to you.
Thanks, Allie. So I'm John Olson. I'm the interim president of the California chapter of Freshwater Science. I'm interim because we haven't had an election. So one of the things we'll talk today, our business meeting will be electing new leadership. But anyways, welcome all to our meeting here. Let me see advance. And I also want to thank our sponsors that helped us provide all the goodies you got there. So Watercourse Engineering, Aquatic Bioassay, NIAD, and Rithron. So Freshwater Science, for those of you not familiar with the society, is a dedicated to basically translating the science of health and vitality of freshwater ecosystems to the public and to each other. We here at CalSFS, we kind of see this as a chance for us to get together and actually interchange ideas and actually pass ideas on to the next generation as we have students come to these events. Freshwater Science is approximately 1,500 members, almost a third of them students, and they basically cover all the possible ecosystem, related freshwater ecosystem related concepts you can think of. So all the pieces are covered by Freshwater Science. We also produce our own journal, which is peer-reviewed, published quarterly by a nonprofit publisher. You can actually apply to get grants if you you need additional assistance actually publishing, or get fee waivers. Other activities for benefits of being an SFS member, you get discounted cost to go to the annual meeting. You get a free subscription to the Freshwater Science Journal, as well as access to numerous professional and student awards. Taxonomic certification program is run by SFS. And you get to basically to interact with a large variety of freshwater scientists from across the globe. We also have our own mentoring and inclusion program for students, both InSTARS and Emerge, for either graduate students or all the way through postdocs. Are encouraged to apply for these programs. They provide funding. You get to students from non-representative minorities to come to the meetings, so they can come and see freshwater science for themselves up close and personal. And one of the reasons we have these California these statewide chapters or regional chapters is basically allows us to have small meetings like this where we can talk about focused California issues, and also allows us to get more people to come to a meeting that may not be able to come to a meeting in, say, Spokane, which is where we're going to be this next spring. So this basically gives us a chance to facilitate our conversations here locally in California. So the annual meeting for the actual society will be this year in Spokane, Washington. If you're able to come, I strongly encourage you to do so. And if you want to get involved, and you want to join CalSFS, if you're not a formal member of CalSFS, you can join SFS by going to the website, and CalSFS is basically costs $5 more. And with that, I think I'm supposed to remind you, so we're going to have a business meeting here at 4. If you are a member, you certainly should be here. If you're not a member or you're just curious, you're welcome to stick around and talk to us about what we're doing in terms of what CalSFS is going to be doing in the near future. And after that, we have our mixer. We'll talk to you more about the mixer at the end of the day, but basically, there's a chance for all of us to get together socially and talk to each other. We'll do that tonight. With that, I'll turn it back over.
Okay, so we realized that the QR code was broken. So if you tried and it did not work, retry. It's fixed now. Okay, so next up, Felicia Walls with the Swamp Bioassessment Program is going to provide some announcements and updates from the statewide program. And Felicia just joined not too long ago, June, this summer, as the statewide bioassessment coordinator. Felicia, over to you. Yeah.
Good morning, everyone. Happy you all were able to make it today. So again, my name is Felicia Walls. I am the new Swamp Bioassessment Program Coordinator, and I'll be giving the announcements and updates today. So starting with our non-perennial stream assessment update, we're shifting our monitoring efforts to focus on identifying and sampling non-perennial reference sites in Northern California. In 2015, the team successfully sampled 31 non-perennial reference sites across 41 visits, but 10 sites were found dry. We now have a total of 50 stations, 50 non-perennial stations, with the goal of 70 non-perennial sites. So we're almost there. Over the past 2 years, nearly 500 potential sites have been scouted, so that's amazing. And PHAP flow data combined with GIS tools are guiding site selection for 2026. Chad Laughlin and Nathan Mack will be giving a presentation during session 1 today. So if you're interested, please join us. All right, we are developing a dynamic table and internal dashboard to track the most up-to-date reference and flow status of sites across California. An internal-facing version is now utilized to support data review, and work is underway to develop a public-facing dashboard. Let's see. Of the remaining sites, it is expected to be completed by the end of the calendar year, so fingers crossed. All right, CMAP is using eDNA collection and sequencing to produce taxa lists for fish, benthic macroinvertebrates. There's a lot going on this screen. I can't see that. And I don't want to miss any important information. All right, so let me start over. CMAP is using eDNA collection and sequencing to produce taxa lists for fish, benthic macroinvertebrates, and algae, and to generate biodiversity metrics that integrate biological habitat and chemistry data. So please attend Lindsay's talk today during session 2 to learn more. Next year, we will begin exploring the feasibility of bird-based bioassessment as a new indicator of stream biological health. We're all excited about that. Birds may also provide a more accessible, community-engaging approach to monitoring. So this approach aims to enhance the bioassessment program while providing a more accessible, engaging way for communities to participate in assessing and protecting stream ecosystems. All right, we've also developed communication materials to help explain key bioassessment concepts such as reference condition and use of biological indices in intermittent and modified streams. The reference fact sheet is under review and expected to be finalized by mid-November, while work is underway on the biological indices fact sheet. If there are other topics that you would like us to interpret for the general public, let's talk. So find me at any point of the conference. SWAP hosted its largest Bioassessment 101 training on September 4th of this year. In Sacramento, we had 27 participants attending classroom and field sessions led by experts from SQUIRP. Led by experts from SQUIRP, CSU Chico, the Aquatic Bioassessment Lab, CDFW, which is the California Department of Fish and Wildlife, and the San Diego Region Water Board. The purpose of this training is to develop the Water Board staff knowledge and skills required to collect and use bioassessment data. We extend our appreciation to all of our presenters, most of whom are in the first and second row today. So thank you all very much, our field leads and participants who contributed to the success of this training, and we look forward to offering Bioassessment 101 in the near future. All right, as part of the program's effort to improve engagement with communities, we've been working on an effort to revitalize and better integrate SWAMP's biological monitoring programs, like freshwater harm from algal blooms and bioassessment and eDNA and CMAP. We want to make sure we're generating accurate, high quality, and accessible biological data that really serves California's communities. As part of this, our team has been focused on building in-house expertise and field skills. We've completed 4 field trainings. So far, an exciting milestone for our program, which has traditionally relied on receiving rather than collecting our own data. These sessions are helping staff gain the hands-on experience needed to better respond to the complex technical questions we're receiving from local communities. And we're not stopping there. More field days are planned with the focus on bioassessment and eDNA sampling protocols, and hopefully good weather is on our side. And that concludes our announcements and updates.
Let's go.
Your favorite kisses. You're welcome to use this.
Good morning. Glad everyone can come. My name is Dan Pickard. I'm with the Aquatic Bioassessment Lab, but I'm also a member of SAFIT. You wonder what SAFIT stands for? It stands for the Southwest Association of Freshwater Invertebrate Taxonomists. I know, a lot to put in one mouthful, I know that. This is our mission statement up here. Basically, the mission of SAFIT, we're the taxonomic backbone to what happens in the bioassessment world. So through standard taxonomic practices, we support SWAMP's mission to look at bioassessment in the state. Let's make sure I'm going. Oh, I did it wrong again, Dan. Okay. So one of the ways we support SWAMP in its effort is to maintain the standard taxonomic effort document. The STE is what us taxonomists consider reliable taxonomic effort on your benthic macroinvertebrate samples. So say Oligoceta, you can't go any farther than that. So in this document, we put that out, show you where you can go with different things. It's been quite a while since we've made a standard taxonomic effort document. And that is because this time, Doug Post, who's listed first up here, went through and he put into a database all the initial taxonomic literature designed around any family or any taxon's name into that database. So someone updating this later doesn't have to do that. So all the literature for all these names is in this database. Huge, huge effort. So big round of applause to Doug if you were here. But then Andy Wren and John Sandberg also in the Aquatic Bioassessment Lab were huge in doing this. The other thing that SAFIT does is provide taxonomic workshops. Now, in the past 5 years or so, we haven't had much in the way of hands-on taxonomic workshops. One of the reasons was COVID, right? Couldn't have a hands-on taxonomic workshop during COVID. So things kind of fell apart. This year, we're pleased to announce that we are going to have one. It's going to be here. It's going to be tomorrow afternoon in room 1710. And one of the biggest changes in the standard taxonomic effort document this year are these taxa. These are predaceous diving beetles, and they have split out, which used to be 2 genera, now into 7 or 8 genera. So it's one of the hardest parts of the key, or of our STE document. And we're pleased to have Doug Post from our lab give this workshop tomorrow. There still are a few spaces left if people want to go to it. just contact me at some point, and we get you set up. So one of the other reasons why SAFIT hasn't been active in the last few years is our acting president, Joe Slezark, passed away this last year. Joe has been part of the statewide bioassessment world here in California for 25 years. He did his undergraduate and graduate work at Penn State. He's 30 years as a professional taxonomist. Mayflies were his favorite things, but just like the rest of us at the ABL, he could do everything. So taxonomically, he could do everything. If you had any samples submitted in bioassessment in the last 25 years, he probably worked on your samples. He's responsible for over 78,000 lines of data in the SWAMP database, personally. He's the co-author of the current SWAMP lab SOP, which I know our lab follows and most other labs follow. And he was the first SAFIT president. So Joe and Rafi and Ken Schiff and Pete Odie were all, the people that really got SAFIT going. And then just personally, I've known Joe for 30 years. He was an avid fly fisherman. He would often, when we lived in Paradise, after he got home, he'd throw his belly boat in the back of his car and go up to DeSable Reservoir, which was 10 minutes up the hill, and fish all night. So if you knew Joe, his laugh was infectious. He could be around the corner and you'd hear that laugh and be, oh, Joe's here, okay. And we had lots of different adventures together. This is me and John and Joe up at Lake Davis in 2007 when we did the species inventory survey before they did the rotenone treatments. So I just wanted to take a minute and just acknowledge Joe and thank you for listening.
Thank you, Dan. Thanks for those kind words in memory of Joe. All right, so we are happy to have Caitlin Boise here with us to provide our keynote presentation. Let me get you your speaker mic situation. Just kind of wear it like a necklace. Caitlin's going to be talking about the Klamath Dam removal and the past year of renewing and rewilding that river and their journey. And before you start, little intro, careful phrasing. So Caitlin Boise is an aquatic ecologist on the Fisheries and Aquatics team at Resource Environmental Solutions. Her work focuses on water quality, data management, and aquatic species conservation as they relate to restoration and mitigation banking projects on the West Coast with RES. And with that, Caitlin, it's all yours. Yeah, so just push the button and it's green, you're good to go. And you can clicker or advance on the keyboard.
Okay. I don't want to throw you under the bus, but it is Boyce, not Boise, just so that when everyone wants to complain about me later, they know how to say my name. I'm really excited to be here today to present a look back at the past year of the Klamath River Renewal Project's restoration journey. And today I'm here to talk about the Lower Klamath Project, which included 4 dams built between 1918 and 1962 across 40 miles of the Klamath River and 2 states, as well as the Klamath River Renewal Project, which is the effort to remove those dams and restore the over 2,000 acres of habitat that emerged from under the reservoirs that those dams impounded. And as much as we love to look at pictures of dams blowing up, I'm not going to be talking about the physical dam deconstruction too much today, but instead I'm going to be focusing on the restoration that has occurred in 2024 and 2025, and some of the ways in which a project of this scale thinks about assessing ecosystem recovery and project success. And I'm focusing on that aspect because RES's role is really as the restoration designer and contractor. We've supported a lot of other efforts along the way, but we are responsible for designing and implementing restoration across these newly unearthed former reservoirs, as well as the long-term monitoring and maintenance to meet some of the compliance criteria for the project. And there are obviously many contributors that have made this project happen, all under the Klamath River Renewal Corporation, which is the dam removal entity, with their client rep McMillan, Camas providing permitting support, and Kiewit Infrastructure West, the ones actually doing that dam blowing up in that picture before, and then us as the restoration contractor. And obviously, none of this work would have been possible without the tribes in the basin spending decades advocating for change, really bringing this project to fruition. None of that would have happened without their advocacy. So just want to remind everyone of that. So we'll step back for a moment, take the high-level view of the project. And the purpose of the project was really to achieve dam removal, a free-flowing condition on the Klamath River, and volitional fish passage. Obviously, there were benefits anticipated beyond that, but that was the high-level goal. And we did that. In 2023 and 2024, Kiewit Infrastructure West completed the deconstruction of the 4 lowest dams. And I'm not going to talk too much about that, like I mentioned, but just to give you a sense of scale, wanted to show some before and after pictures. So this is J.C. Boyle Dam and the reservoir it impounded. You can see it's partially earthen, partially concrete structure. Before dam removal and after. And then CABW number 1 dam and CABW Lake upstream of it. CABW was the largest dam, entirely concrete. And the Klamath River flowing through CABW Valley after dam removal. And finally, Iron Gate Dam, which was an earthen structure. And if you were a fish swimming upstream in the Klamath River before dam removal, this was the end of the road. This is where your journey stopped. But then less than a year later, it's an open gate to hundreds of miles of additional habitat. So all of that happened in 2024. And before drawdown of the reservoirs was even complete, before dam removal had even happened, we began restoration. And this included sediment evacuation in some key locations and some of the tributaries in the former reservoirs, and removal of some anthropogenic debris that might cause fish passage barriers. So think things like old bridge abutments that were just lingering under the reservoirs for over 100 years. We also began revegetation to take advantage of the residual moisture in the reservoir sediments. So this included a partnership with the Yurok tribe and the hand seeding and helicopter broadcast seeding, as you can see in this picture, hopefully, as well as container, bare root, acorns, all planted in the former reservoirs. And this work really began several years before dam removal with the collection of close to 100 species worth of seeds from the basin, so native adapted plants that were then propagated. And between the seeds we collected and the propagated plants, we had over 19 billion, with a B, seeds to work with on this project. And as of this spring, I think we've applied like 100,000 pounds or so to the landscape, so a huge reseeding effort. And finally, a lot of large wood loading within some of the tributaries to really try to jumpstart restoration and create some complex, good habitat for fish as they return. So that was all 2024. Like I mentioned, before the dams were even out, really trying to get restoration underway. And then in 2025, we really began our high priority tributary restoration in earnest. And I'm not going to talk too much about Spencer Creek up in Oregon, mostly because Spencer Creek had a really small segment under the reservoir and it was already pretty well connected to its floodplain. So really all we did out there was apply some seed and place some large wood in the creek. But in 2025, we performed more targeted restoration on Camp and Jenny Creeks in Iron Gate and Beaver Creek in CABW. So let's look at what that looked like. So the Camp Scotch Creek complex was our largest segment of stream, over 8,000 linear feet of restoration and over 190,000 cubic yards of earthwork, and a lot of large wood placed in this creek. You'll notice at all of these sites, these tributaries had flown into reservoirs where they had hit that slack water and distributed a lot of sediment into these deltas. So a lot of the work that we did was really to make these creeks be more connected with their floodplains. And that involved a lot of earthwork. So this is the confluence of Camp Creek with the Klamath River before construction began. And this is after. You can see they've pulled those banks back to really create kind of an inset floodplain, added some large wood, and there's a couple off-channel features there as well. Work at Jenny Creek was really similar. It was a little bit smaller segment of stream, almost 3,000 linear feet with 52,000 cubic yards of earthwork. Also a lot of large wood placed here, and then a large off-channel pond built. And this bank that's in this picture has whole trees embedded in it, both to provide stability to the bank, as well as complex habitat and cover for returning salmonids this year. Hopefully the scale of this is a little bit clear with that large piece of equipment and the whole trees in the bank. So this is Jenny Creek looking downstream towards its confluence with the Klamath River. A fun side note is that prior to the winter of 2024-2025, this was a single thread channel through this reach, and then Jenny Creek had a flow of about 2,000 CFS, and it reorganized itself into a multi-thread channel. So we had to adapt our plans a little bit when that happened. But you're going to see in the after picture that the banks, especially near the confluence, have been graded back to provide some more floodplain connection. There's large wood, and then additionally, the most obvious change is a large pond kind of in the bottom left-hand corner of the picture. And like I mentioned, Jenny Creek flows can get pretty high in the winter, so that pond is going to provide an important velocity refuge when flows in the creek are in the thousands of CFS. Beaver Creek had some similar work done, over 6,000 linear feet of stream with 55,000 cubic yards of earthwork and a lot of large wood. But in Beaver Creek, we actually built 28 beaver dam analogs across the lower half of the restoration site. We're going to talk a little bit about why that is. So this was a really simplified in size channel, and the original plan had really been to do similar work as the other creeks, sort of grade down the surrounding banks to let the Beaver Creek access its floodplain. But we had this really wide valley segment to work with. So instead of bringing the banks down to the creek, we decided to bring the water up to the valley and install BDAs throughout this lower reach. And this is what that looked like. You can see much messier, a lot more material in the creek, more of a meandering stream, and water levels really responded quickly. And this is creating some really good complex habitat that we think is going to be important for fish as they return. So finally, this is a big project. It's hundreds of miles of river, a lot of moving pieces, time and space are huge. So how do we document ecosystem recovery and project success given that scale? And the main question here is, how do you capture change on a landscape scale? And if it isn't already clear, photo monitoring has been a big part of that. We've been documenting things like locations that might need adaptive management or places where our performance of vegetation is good, but also just telling the story of the project. And we've been doing that with almost 200 ground and drone-based repeat photo points. So someone going to a site every month or so and taking a picture. We've also had time-lapse cameras installed, which have been great for construction and deconstruction of the dams. And we always say that taking someone to a site is the best thing you can do to show them the project. The next best thing is showing them a high-quality picture or video of what's happening out there. And so I mentioned that while the goal of the project was a free-flowing Klamath River and dam removal, there were anticipated to be a lot of additional impacts and benefits of the project. And one piece that we're trying to document is the water quality improvements that have followed. And we're doing that with a pretty expansive water quality network. This includes stations that were already in place and run by USGS, as well as some that were installed specifically for the project. And it covers over 230 river miles. And the main aspects of it are 9 telemetered stations that are collecting continuous data and telemetering it in near real time. And then 9 grab sample collection locations. So where we're collecting biweekly suspended sediment concentration samples and monthly samples for other parameters. And this, for the project and compliance purposes, has been happening since 2023. So you can imagine that we have a lot of data that we're dealing with, hundreds of thousands of continuous data records and thousands of grab sample analysis results. But one piece that I really want to pull out and talk about is water temperature. So one of the anticipated benefits of dam removal was the removal of these reservoirs, which acted as large heat sinks. And they really prevented water temperatures from fluctuating naturally, both on a daily and a seasonal timescale. And what I'm going to show here is the daily minimum and maximum temperature at the station immediately downstream of Iron Gate Dam in the year pre-dam removal. So hopefully what's clear from this plot is that it didn't vary a lot. The most, maybe 2 degrees Celsius, but in the winter and fall, half a degree or less Celsius of daily temperature variation. And in 2024, that changed really dramatically. We had 3, 4, 5 degrees of daily temperature variation almost immediately after the reservoirs were drawn down. And we saw something really similar in 2025 with much wider range in daily variation of temperatures. And I also want to highlight that despite temperatures being warmer sometimes during the day, we're still seeing really cool temperatures at night. And I mentioned seasonal scale temperature variation. And we've been able to document a change in that as well. So the reservoirs would delay the water temperatures increasing in the spring, prevent them from decreasing naturally in the fall. And with the reservoirs gone, we've seen earlier warming in 2024 and 2025, and some earlier cooling in the fall. And temperature provides really important cues for fish, both in terms of spawning and outmigration. And it also just really impacts what habitat they can actually access and use. So the return of this more natural temperature regime is going to have a huge benefit to the Klamath River. Another piece in the puzzle of documenting success is recording the change in species assemblage. And we're doing this through what we're calling a molecular time capsule and a series of eDNA collection efforts. I know we're going to hear a lot about eDNA later in the day, but in case you're not familiar, it's DNA shed by organisms that's collected from the environment rather than from the organisms themselves. In our case, in water samples. So we collected in 2023, 2024, and 2025 over 1,000 water samples from 45 locations across more than 70 river miles. Really large effort, using a before-after reference impact design. So we have control sites within the basin, reference sites outside of the impacted area. And the goal was really to create a snapshot in time of what the biodiversity in the river looked like before, during, and after dam removal. And additionally, to make this data available for contemporary and future research into what the species assemblage looked like. And if you want to learn more about this effort, my colleagues recently published an article in Scientific Reports. The title is there. You can learn more about what they actually collected, what they've seen, especially in the pre-dam removal year and the dam removal year. Finally, we're of course tracking the return of salmon. And our primary objective as RES is the 21 miles that used to be under a reservoir. So we are responsible for ensuring that fish can pass through these 21 miles of habitat and reach the hundreds of additional miles of habitat that they now have access to. And we're trying to track novel returns and occurrences of fish on this website here. You can check it out, but the population monitoring for these species is being done by a wide variety of groups who are using different approaches like radio tagging, spawning and carcass surveys, eDNA, sonar stations throughout the basin. And in 2024, we documented fish traversing the Iron Gate Dam footprint, which is the lowest dam in the system, just days after in-water work for dam deconstruction was completed. A few days later, they were in Jenny Creek, like these fish you see here. And 2 weeks after the final in-water work was done for dam removal, fish had passed through the entire project footprint and made it up to Spencer Creek in Oregon. And it happened more rapidly than we thought, and we were so excited to see that. We couldn't imagine anything better until 2025 happened. And with better water quality and river conditions, Chinook are returning earlier and encouraging numbers at the start of the spawning season. We don't have final numbers yet because we're still in the spawning season, but fish returned to Jenny Creek about 3 weeks earlier than they did last year. They were in Oregon up in Spencer Creek about 3 weeks earlier than last year. And the most exciting news of all is really that on the week of October 17th, Chinook were detected in the Sprague and Williamson rivers. So they have passed through the entire Klamath River, Upper Klamath Lake, and made it into the upper basin tributaries that they have not been able to access for over 100 years and are now spawning in those tributaries. And there's still a lot of work to do in the basin. There's a lot of monitoring for us to do on the Klamath River Renewal project. But the goal of this project was to restore volitional fish passage. And the rapid return of these fish is just a reminder of how far this project has already come. So if you want to follow along with the restoration, there's a QR code here for a story map that's being updated on a regular basis. And I'll leave you with a couple of the 2025 returning Chinook and Jenny Creek who are probably spawning as we speak. Thank you. Do I have time for questions?
Yes, we have time for questions. Any questions?
Just so you know, you're getting a lot of love in the Zoom.
Oh, thank you. I appreciate that.
On.
Hey, I'm just wondering about some of so your primary goal was the volitional passage. Among the other goals, which ones have you prioritized trying to assess, and how are they looking there?
Yeah, the second big piece is stabilizing reservoir sediments. So there was sediment that had accumulated in the reservoirs that obviously did not get flushed out of the former reservoirs. And so we're tracking that mostly using our water quality station, tracking sediment load, seeing how storm events or how the reservoirs respond to storm events. And then revegetation is a really big piece. And that's all happening with standard transects and plots that they're going to be tracking species assemblage and percent cover and all of that throughout the next 5 years, I believe.
Speak really right into it.
Thanks. My question is about native mussels in the system and what you guys are doing to look at native mussels and recovery, reintroductions, anything like that?
Yeah, we aren't specifically doing any of that work. I believe there was an effort before dam removal to transplant some of the mussels further downstream so that they weren't too impacted by drawdown. I believe that was work that Xerxes Society was doing. So I think they're doing a lot of the work with mussels in the basin. We will be tracking that with eDNA. We have those samples, but we're not specifically doing any of that work beyond supporting what Xerxes is doing. Yeah.
Hi, great presentation.
Thanks.
I'm just curious with the revegetation efforts, is there any worry that any invasives would get in there, and what are some preventative measures you guys are adopting or doing?
Yeah, it's definitely a big worry. We've spent the last couple years doing a lot of treatment around the border of the reservoirs. So weeding, trying to get star thistle out of there. That's one of the big invasives that we have around the reservoirs. There is an IEV program that they're following along with the tracking the natives. We're tracking IEV and we're doing our best to catch it early. Our hope is that with the reestablishment of natives rapidly recolonizing the reservoirs, that it's less of an issue, but it's definitely something that we're tracking and focusing on just rapid response. Yeah.
We have a question / comment in the Zoom. This is the inspirational story of the century. A lot of us need. Great presentation, a lot of information. Is there a plan to build open data backend dashboards for the complex data sets?
Yeah, a lot of the data from our water quality networks is available online. Like I mentioned, a lot of it was USGS stations and then the stations we installed for the project were set up as USGS stations, so they're all real-time online. I'm not sure about all of the vegetation data. Eventually, it may become available and open to folks to use. We're always open to folks reaching out if you want the data and you want to do something with it. Just reach out, send me an email. We can work on that. But there is a lot of information being collected, and we do not have the resources to assess what all of it means. So definitely reach out.
One of the sites, you said you put in beaver dam analogs. I think that's because there isn't enough wood for beaver to do it on their own. Are there beaver in the watershed that will be?
Yeah,
building their own beaver dams?
we hope so. We hope they come in and they look at what we did and they're like, this is wrong, and then build their own. Yeah, that's really the hope. And yeah, exactly. Yeah, we do anticipate that they will recolonize. There are beaver in the watershed. We've seen them. We've seen signs. So yeah, really excited about that.
We have another question in the Zoom. With all the different partners monitoring the salmon, is there some coordination with a standard monitoring protocol, or are partners focused on various items?
Yeah, most of the population monitoring is being run through CDFW and U.S. Fish and Wildlife, so it's being standardized by them, and with many partners conducting the actual on-the-ground work using their protocols. So that is all pretty standard. We're supporting some of that. The Karuk tribe is supporting a lot of that work. But yes, that is all standard.
Yeah, just a quick question about
Wait for the mic, please. The internet needs to know.
Just a quick question. What are the problems they had with it when they removed the Elwha Dam up on the Olympic Peninsula? It took many, many decades of planning. Was they had a big sediment plug that went down the river. So you mentioned that a little bit. Anybody following up on that? You concerned about that or monitoring that? Thank you.
On the Elwha Dam, or this having a similar issue with the Klamath dams?
Well, I'm asking, yeah, are you monitoring that? Are you concerned about the Klamath Dam having the same sort of impact?
Yeah, so most of the sediment that was impounded by the Klamath dams was dead algae. It was really super fine material from all of the harmful algal blooms that had happened on the lakes over the years. And it was really easily mobilized downstream. So a large proportion of it made its way out into the ocean. And one of the mitigation measures for the project was actually tracking whether that all ended up in the Crescent City Harbor, because there was the potential for currents to carry it there. That didn't happen. So we don't actually know exactly where it ended up, but we do have some measures around tracking kind of how degradation of the reach or aggradation might have occurred in the reach downstream. And we haven't seen a lot of that happening. So yeah, very different sediment composition than what was in the Elwha.
Well, thank you.
Yeah.
Boys. This is thank you. Thank you so much. Okay, so our next talk is going to be by Chad and Nathan on the swamp bioassessment and intermittent streams. They're going to provide a statewide update and next steps. Let me give you your fancy intros. So you'll be talking first, right? So Chad is a senior environmental scientist at the San Diego Regional Water Board and a member of California's Swamps Statewide Bioassessment Workgroup. Chad oversaw implementation of a program to evaluate bioassessment in the San Diego region intermittent streams in 2013. And then Nathan is a senior environmental scientist with CDFW's ABL lab. He started with ABL in 2008 and has moved between CSU Chico and State Water Board and CDFW, but always focusing on bioassessment. And
All right. Thank you, Ellie. So this is a talk that Nathan and I are going to be co-presenting on, basically giving an update on intermittent streams and how they're being assessed by swamps statewide. And like it was mentioned earlier, this is not just Nathan and I, but it's really a group effort of the state swamps bioassessment work group, which includes lots of entities, including ABL, SQUIRP, I'm going to forget somebody, the regional boards, Moss Landing Marine Lab, and I'd be remiss if I don't mention all the swamp OEMA folks, including data management, contracts, infrastructure, and all of that. So to start off, I'll give a little bit of a grounding introduction and background. A lot of people, this is probably review, but just in case, the state of California developed a statewide index of biotic integrity, or an IBI, for stream bioassessment back in 2016, called the California Stream Condition Index, which we affectionately refer to as the CSCI. It uses benthic macroinvertebrates as the biological indicator. And what's unique about the CSCI is it's a modeled index. California is a really diverse state with a lot of diverse stream types. So what that means is that basically a sampled site's biological expectations are based on reference conditions. It's grounded in reference condition. But because of stream diversity statewide, that reference condition is actually based on a stream's natural attributes, non-anthropogenic, things like elevation, watershed size, precipitation, and geology. And that sets the reference sites that a site is compared to for scoring using the CSCI. However, we do know that these reference sites were not picked based on natural flow conditions. And that wasn't some sort of willful ignorance. That was really just a matter of bioassessment sampling is done in the spring and summer during a proper index period when streams are flowing. And so that's led to a management question that's come up is, does the CSCI itself work in intermittent streams? And this has come up statewide. It also came up in Region 9 for development of our biological objectives. And we spent pretty significant time and effort addressing it in Region 9. And now we're expanding that work as part of the statewide bioassessment work group across the entire state. So the question comes up is, what is an intermittent stream? Well, for the purposes of this project, an intermittent stream is a stream that will flow in the winter or spring for enough time to form a benthic macroinvertebrate community that can be sampled using the swamp SOPs. And typically it will dry up in summer, late summer, early fall. Maybe a better way to describe it is, what isn't an intermittent stream? Well, perennial streams, which always flow, have surface flows. And for the purpose of our work, ephemeral streams. Yes, they're intermittent streams, I know. But really for us, ephemeral streams aren't targeted. They only flow in direct response to storm events, usually for 48, maybe 72 hours following a storm. And so we're really looking at these intermittent streams on the left. And Nathan's going to talk a little bit more about identifying those for sampling. That's what we're looking at. So the approach that we're taking on a statewide basis actually is mirroring the approach we took in Region 9 for biological objective development. And we're taking a two-pronged approach to try to get at this question. One is to determine the flow status at CSCI reference sites. So the sites that were used in the CSCI development data set that are used for calculating CSCI scores, are they intermittent? Are they perennial? What does that look like and where? And then secondarily, identifying and sampling new intermittent reference sites, including sites that weren't used in the CSCI development, to basically see how the CSCI is working in intermittent sites around the state. And I actually put a little sample and sampled because we do know that we've got a pretty big data site for reference sites. So we can go back to existing reference sites that we think might be intermittent, document their flow condition, and use that data. So I'm first going to go over approach one before handing it off to Nathan to talk about identifying new sites. And this is a map that shows, probably a little hard if you're reclusive and sit in the back. Basically, it shows all of the known flow status at reference sites that are used in the CSCI itself. And I'll point out a couple of things here. It's a little hard to see, but we have intermittent in green circles, perennial in blue, and if we don't know the flow status, it's a question mark. So first thing you'll notice is that in Southern California, which is Region 9, Region 8, 7, Region 4, from a water board perspective, we've got a lot of knowledge on flow status. And that's because in Region 9, we did a lot of work for biological objectives, looking at CSCI reference sites to see if they were intermittent or not. Region 8 and Region 4 was also funded via SWAMP, looking at flow status. And you'll see, so in general in Southern California, we've got a lot of intermittent sites that were used in the CSEI development and a smattering of perennial and some unknown. However, once you go outside of Southern California, it tends to be a different story. You'll see a few intermittent sites in the Bay Area, Central Coast, a couple in the Sierra, but mostly it's perennial or unknown, especially in some of the northeastern part of the state and the MODOC. North Coast is predominantly shown as perennial or unknown. So I'm going to skip the next slide in the interest of time and go straight to Nathan. Thank you, Chad. Yep.
Thank you, Chad. So I'll go back to Chad's slide for just one sec. Because this is going to be my lead in here. You can see that from just looking at this map briefly, we are lacking intermittent reference sites throughout most of the northern parts of the state. So to help answer the question if we can use CSEI throughout the state as a whole, we're going to need to establish many more sites in like North Coast, MODOC, Lahontan, Western Sierra, Central Coast, and interior Chaparral ecoregions. This is a very large area to cover. And these sites, it's going to be challenging because these sites need to be both intermittent and in reference condition. And since we're concerned with CSEI, our criteria for reference condition will be the same as the criteria used in CSEI development. The table on the right is a list of the metrics that define reference condition. Most of them are measures of human disturbance derived from GIS data, with the exception of one circled at the bottom, W1 Hall. That is a reach scale disturbance measure. It comes from the bottom of swamp paperwork. There's a human influence section down there, and it's a combination of all those metrics combined. Chad touched on what we're looking for. We want an intermittent stream, so you can think of hydrologic state as a continuum with very dry streams on the ephemeral side and wet streams that are unaffected by periods of drought on the wet side, the perennial side. And we're looking for a sweet spot right in the middle, these regularly flowing intermittent streams. So a perfect stream for this would something flowing between like 6 to 9 months, but would still dry in most years. Chad mentioned we were looking for sites we've already sampled that were part of the CSI development. We're also looking at all reference sites that we've already sampled, hoping that we have maybe sampled some intermittent streams previously. We're confirming hydrologic state with looking at water level logger data that may have been collected or notes from previous site assessments. And we're also prioritizing previously sampled reference sites for follow-up visits during the dry period of the year using PHAB data associated with those sites. So if flow is too low to measure, there's a QA code for that. If the max discharge was very low, we're going to prioritize that site. And if the predominant flow habitat was mostly glide or low flow habitat types. We're continuing our outreach campaign, talking to researchers, land managers, and anyone that's just kind of generally in the know about streams in their region. This is a brief list of people that we've worked with so far, and their firsthand knowledge of their streams has been instrumental in getting this first group of intermittent reference sites together. So while those first two approaches have been fruitful, they're not going to get us all the way there. We're going to need many, many more sites to both screen for reference condition and follow up with a site visit. We've been coming up with these points by handpicking locations on maps. We've created thousands of points by looking at the intersections of roads and NHD flow lines. Those have been useful since we have some confidence about being able to reach those locations physically, given that they were established based on a road being present in that area. And then when we do establish a sampling reach there, we would place it upstream of that intersection to minimize human influence. And then since it's unrealistic time-wise to screen every potential watershed by delineating and then running the reference screen across it. We created a rudimentary reference screen. The most common metrics from the reference screen to push an intermittent site out of reference condition are code 21 from NLCD data, that's open space developed land, road density in a watershed, and presence of an active mine. So we took those 3 metrics and used HUC-10 polygons as surrogate watersheds and applied those metrics to those polygons to create like a map of regions where we'd have a higher likelihood of finding reference sites. And we've been using that map to help guide office-based decisions on where to start looking for sites. And while field crews are out exploring watersheds, they'll use that to help make those decisions for themselves in the field. And when I say field crews exploring watersheds, I mean Allison Furler and Connor Stewart are 2 very dedicated biologists will conduct swamp bioassessment on statewide projects through all of spring and summer. And then as soon as sampling season ends, they get to start, I feel like I'm getting a reverb. They get to start recon season as well. And so they're out in the field for close to 9 months a year now. So I think the overall success of this project will be largely due to their hard work. So while people are out in the field, what they're essentially trying to do is guess from a single visit to a dry stream in the fall what flows are going to look like come springtime. To help educate their guests, they are using physical indicators of these streams. So things like watermarks on rocks, riparian vegetation along the stream corridor, dried algal mats are helping them make these decisions, along with relying on years of experience conducting stream surveys. If they think that one of these candidate streams will make a good intermittent reference site, they'll install a water data logger. For this project, we're using onset tidbits and recording presence or absence of water and water temperature for a 2-year period. So we've been hunting these intermittent sites for about 2 years now. While in the field, we're using a mixed method of going to predetermined sites where we already know about their GIS reference condition, and field crews only need to confirm the reach scale human influence and hydrologic state and accessibility. And then while they're out going to these predetermined sites, they're also exploring the nearby watershed and hopefully coming up with new ideas of places that we didn't think about before they went out for the week. Mixing these 2 methods together has proven to be much more time effective, and crews are able to visit up to about 30 sites in a week. So this map is a summary of what we've been doing for the last 2 years for non-perennial recon. Red dots are points that were either inaccessible or crews thought there was too much human influence at the reach scale, so they wouldn't end up passing reference screen later. Orange dots are too dry, blue dots are too wet, and green dots are our sweet spot again, places that we think will have flow for like 6 to 9 months. And then the green dots with circles around those also pass reference screen and have a water data logger installed. So this effort has produced 58 stations that we have BMI data on so far. But that is not accounting for sites in the Bay Area. Given the sufficient spatial coverage and index performance there, which Chad will touch on again in a second, we're not prioritizing trying to find new places. For 2026, we have 30 new stations from fall recon this year that we plan to sample. And these look like some good numbers. I still think we'll need more for fall of 2026 and beyond. So I'd like to take this opportunity to request of all of you suggestions. We use Survey 123 in the field for submitting data internally, but we also have a public version of that available for anyone to scan onto their phone. There's some QR codes in the back of the room. And if you don't want another app on your phone, I get it. You can just send emails to me. All I really need is GPS coordinates, and a couple photos would be great. And if for some reason you forget my name, Nathan Mack, within the next few weeks, and you find a great intermittent stream, you can just send it to the generic email address, bioassessment@wildlife.ca.gov. And I'll turn it back to Chad.
All right, I know we're short on time, so this is really the, oh man, there we go. Okay, so initial results. So this is really the last real slide we have showing the results so far from the effort. And we're presenting it two ways here. On the right, we have a map, and the left, it's box plots for sampled sites by regional board. Included the map because not everybody's regional board centric, I understand. Start with a map. The map basically shows intermittent reference sites that have been sampled and their scores. Do they meet the 0.79 threshold that's used for the CSEI to show streams being impacted or not? Red triangle is no, and the blue is yes, or I'm sorry, the blue is yes. And so if you look at the map, you can see that in Southern California, we have pretty good representation of intermittent reference sites and a lot of high scoring sites. You can see a lot of sites in the Bay Area and the smattering that we have across the state. If you want to look on the left side, which shows box plots by a regional board and California Stream Condition Index scores on the y-axis, the dashed line represents that 0.79 threshold that's used for documenting impacted condition. On the far left, we have the development data set. So that's the distribution of reference sites used in the CSCI. And then we have regional boards from left to right. I'll point out that Southern California, we grouped them all together based on the density of sampled sites. And that includes regions 4, region 6 at the very southern end of the region, because it's a really long region from north to south. Region 7, 8, and 9. And then also on the x-axis underneath, we have the number of sites and the number of samples. So for example, in Southern California, we have 110 sites, 246 samples. Now, if you look at the Southern California score distribution, it's pretty well matched with the referenced, with the development data set from the CSCI. And if you look at the mean's a little depressed, but the distribution is good. The 10th percentile is actually good. So we're very confident in using the CSCI in Southern California because of the intermittent reference sites that went into the development data set and what we see from sampled intermittent sites. If we move to the next highest density, which is region 2, the Bay Area, 41 sites, 63 samples. It's a little bit different. The distribution is a little lower, so we got a lower mean, lower median. However, if you look at the 10th percentile for those sites, it's actually a little bit lower, but if you run some percentile stats on it, we don't see a significant difference at this point, but it is a little lower. It's a different story around the rest of the state. Unfortunately, region 1, we've got 5 sites and 8 samples, so really not a lot of samples there. Region 3, 4 sites, 18, 6 sites, 7 samples. We see a lot of spread. We see a lot of sites that score really well. We see sites that don't. And so what we want to do is, as Nathan mentioned, is try to fill these data gaps. So our real next steps here are going to be identification of new sites, collect more data, both inter and intra-annually at these sites, and where the CSEI is not working and is working, explore why, and try to figure out what to do going forward. So thanks very much for listening. Happy to take any questions, if allowed.
We're gonna, for the sake of time, we're gonna start with the question in the chat because we're between you and break. So how will focusing on the 6 to 9 month sweet spot of intermittent flow influence the utility of the model to evaluate biological conditions at intermittent streams that only flow for 3 months?
So that's a good question. It is more challenging to sample these sites, but it's definitely feasible to do so. As we know, a lot of impacts happen in streams that are higher up in the watershed. So it is important to document those impacts because they do have an influence on what happens downstream. You just have to be better at planning and doing your due diligence ahead of time, as Nathan well knows.
Yeah. I would add that streams that are flowing for less than 3 months would likely not develop the benthic communities that we would find within like the ODE model of the CSCI. So it may turn out that those would just be by default non-applicable.
Yeah, in Southern California, we used 4 to 6 weeks of flow at a minimum to sample. So that's something we can look at in other parts of the state.
Hi.
John.
Hello. I've got a question about determining the length of flow and the water table. And I just went out in the field with Dan and we put some water detecting onset detectors out there. Have you thought maybe possibly not doing it for every site, but drilling wells to measure the water table height and change throughout time?
That is a great idea. I'd love to do that, but the cost involved is something that's been prohibitive for us.
So I think just like the highest level and the lowest scoring CSCI, because I noticed on that box plot, the dots are actual measurements, right? Actual sites. Well, the range was from excellent to
Pretty
below.
bad. Yeah. Yep. So yeah, no, that's something I think would be great to do.
Because I'm a taxonomist and study aquatic insects, I also know that they could be living and growing
In the
eggs
hyporheic,
too underwater, under the surface of the.
We've got another question in the chat. What are your next steps to better understand the why for regions 1, 3, 5, and 6?
So I think our next steps, as I mentioned, are to identify more sites because we really don't have a lot of sites, and to sample those sites over a couple of years, both a couple of times within season and over a couple of years on a year-to-year basis to look at, are we timing the sampling right? And how do these communities look over time on a year-to-year basis?
And to add to that, for 2026, we're going to prioritize resamplings, non-perennial sites that were sampled in 25 that had lowest CI scores, and possibly a little later in the index period before they dry in case we sampled before communities had a chance to establish.
Chad, due to Sigma, there's a lot of mapping of GDEs, groundwater dependent ecosystems going on. Are you kind of tapping into any of that data that might help?
See it again?
Groundwater dependent ecosystem mapping, because of the Sustainable Groundwater Management Act. There's a lot of mapping going on of GDEs that might be a good source of data.
Yeah, I have not looked into that, but that is exactly the type of thing I was hoping to get out of this conference today with some new ideas. Thank you.
Had some discussions with USGS folks about groundwater influence, but yeah, I just thought of it.
We want to make sure y'all get time for a break. You're already 5 minutes out of your break time. So you only have 15 minutes left. There are no more questions in the chat. I saw some hands in the room. Just like hound them during the break, So come back in 15 minutes.
Thank you.
All right, for the sake of time, we're going to go ahead and get started. Our first speaker for session 1 is Dr. Christina Yoshida. She received her PhD from UC Berkeley. She is now an environmental scientist at the San Francisco Bay Regional Water Quality Control Board. She's been in that role for about 10 years now. She manages the Surface Water Ambient Monitoring Program, where she develops water quality habitat, tissue, and bioassessment monitoring projects. So can you guys help me give Dr. Yoshida a warm welcome?
Hello. At least we got most of you guys back. Thank you for that opening. My kids are like, you're not a doctor. You can't fix anything. Trying to fix the creeks. Oh, wait, the other way. Oh, that's kind of, oh. All right, cool. So yeah, I'm Christina Yoshida. I'm the swamp coordinator. I also work on the integrated report at the San Francisco Bay Water Board. Here's a picture of me collecting some of the data that I'll be talking about in one of our intermittent streams in the Livermore Valley. So a quick overview of my talk. I'll talk about some of the data sources, regional overview. I'll talk about some of the conditions in our region and how flow and channel type impact them, some of the multiple environmental variables that are influencing our communities. I'll discuss a little bit about interannual variation in our streams, and then some recommendations on how to collect and use bioassessment data from our region based on what we've learned. Okay, so I can skip this slide because we've already talked about what bioassessment is. It's how we've collected our samples, and the CSCI score is how we assess them, and that there are thresholds for the CSCI and how we label a site either impaired or unimpaired. I also want to acknowledge that there was algae that was collected at these sites, but I'm only going to focus on the aquatic invertebrates or the BMI data. All right. Right, so for this talk, I'm going to be reviewing all of our bioassessment data in our region. I've been here for 10 years and I've happened to avoid having to give this talk for so long. And we have so much data in our region that it's about time that we share this. And luckily, SCORP did a report for us this year summarizing our data. So I'll be heavily using that in this talk. But I'm also going to reference a report that was done by our Bay Area Stormwater Collective. So this is part of our stormwater permittees that were required to do bioassessment in 5 of our urban counties. And then I'll also briefly reference an SFEI report that was focused on one of our counties. Okay, so an overview. We've collected data for over 25 years in our region. We have over 2,500 samples in our region. That's quite a bit for a smaller region with about 1,500 unique stations. These sites range throughout our water board region, and you can see the boundary of that in the map. And these sites were collected by a wide variety of folks. About 20% or so were collected by swamp, regional, and state. About a quarter of them by our stormwater permittee program. And then a big chunk of them, almost 50%, was collected by our pre-stormwater permit programs. This is prior to 2012. And again, these all followed the statewide bioassessment protocol, although the older ones were using an earlier method that didn't include algae or PHAB. Okay, so again, more general overview about most of our sites have been non-reference sites. You know, the Bay Area, it's pretty heavily urbanized, but we do have at least 95 sites in our region that meet the statewide reference criteria. And about 36% of our sites are intermittent. Before I get into our stream, the overall conditions of our stream reaches, I quickly want to talk about thresholds. So we talked about this just right before the break, but the CSCI was developed to not discriminate by flow type. However, we've known for many years that our region's intermittent reference streams score lower in the Bay Area. So in this plot, you can see our reference perennial streams score at about 1.0. That's what we'd expect. But our intermittent streams score at about 0.87. This median's been about the same for the last 10, 15 years, even though we've added multiple streams since then. All right, so since our intermittent reference streams are scoring lower, we are arguing that we should have a threshold that is lower to assess our intermittent streams. So in a 2024 report, SCORP developed an alternative threshold for Northern California. So this includes Region 1. And they proposed an alternative threshold of 0.61 compared to 0.79 to consider a creek or a site impaired. As you've heard from our previous talk, this threshold may change as we gather more data and evaluate more. So for the rest of the talk, I will use this lower threshold of 0.61 for intermittent streams when defining stream status, such as impaired or unimpaired. All right, so here are the overall results of our region. As you'd expect, again, it's highly urbanized. Our mean CSEI score is 0.63. About 64% of our sites are considered, or of the sites that were sampled, are considered impaired. However, on a positive note, 25% of our streams were likely intact, which is pretty exciting. And we have some lovely streams still, even though most of them are not in the greatest condition. Of those unimpaired streams, most of them occur in the upper watersheds. So these are in protected parks, whether run by the state or the county, or above reservoirs that are managed by water agencies. And they range a wide diversity of land use types. Most people are surprised to know that we have lovely redwood forests just an hour away from San Francisco, but our reference streams, our unimpaired streams, also are found in coastal scrublands and oak woodlands, like in Mount Diablo. Our impaired streams, which are the majority of them, are usually found in the lower watersheds. So these are urbanized areas or areas that are highly grazed, have altered channels, or altered flows. One challenge with our unimpaired streams are a lot of them have really high quality reference level stream sections upstream of them. And so it makes it challenging when designating water bodies as impaired, since biological condition can really vary along a longitudinal gradient. All right, so we already quickly talked about flow, but I wanted to show the full plot that shows our reference and non-reference streams split by perennial and intermittent. So as I already showed you, our intermittent reference streams score lower, but if you look at our non-reference streams, they're roughly the same and not significantly different. And you can see in the red in the box showing the means, the difference between perennial and intermittent for the two types. Okay, so now we've talked about flow. Channel type also matters for our stream community conditions. So lots of streams in our region are channelized to varying degrees. Most are in urban areas and were designated or designed for flood control. One challenge is that channel type hasn't been part of the bioassessment field protocol, so most of our data doesn't have this information. So in the next few slides, I'm going to show 3 different studies with different data and classification schemes. These classification schemes aren't meant to be like these in-depth geomorphic categories, but rather useful categories for interpreting bioassessment data, for setting realistic expectations and regulatory action and restoration goals. So this was conducted by our stormwater program, again, in these 5 county urban areas. And luckily, they collected channel type. And so they divided into 3 different categories: hard, soft but modified, and natural. They found that the hard channel streams and most of the soft channel streams were impaired, and that channel type was a significant indicator of biological condition. Then we had a report that was done by SFEI, and they focused in Santa Clara County. It's one of our largest counties. And also they had a really nice GIS layer that categorized all of their channels in the flood control channel area into 20 different classes. That's too much for us. And so SFEI narrowed it down to 5. And we found again that hard, mixed, and soft, so these modified channels, scored significantly lower than the unmodified channels. However, there was no difference among the modified channel types. So next, we worked with SCORP recently and used the same data from Santa Clara County, but instead used the 3 different classifications: natural, soft, modified, and hard. In the meantime, our swamp team actually went out and ground truth all these sites because we saw that there was quite a bit of error in the county data, so be careful when using those. And yeah, you can see that soft and hard or modified channels scored lower. However, if you look at soft modified, there are still some sites that were performing fairly okay and above our threshold. Now you might think, oh, this is exactly what I expect. It's not that interesting. But the importance of doing this analysis for our agency is to show realistic expectations of stream health in our channelized streams. Like I said, most of these are in urban areas. They're for flood control channel. We can't just turn them back into natural flowing streams. So we need to have, again, realistic levels that we can provide for our permittees when they're wanting to do some modification or for goals for setting for restoration in these areas. Okay, so now that we've talked about channel and flow type, next I'm going to show the results of the importance of multiple environmental factors on biological condition. Again, I'm going back to a previous report by our stormwater permittee program. They used a random forest model to look at multiple variables at once, and the model outputs provide an ordered list of importance of the explanatory variables. They used 26 different variables, land use at multiple scales, habitat, and water quality that were collected at the same time with the invertebrate data. And their final model showed that 5 different variables explained about 67% of the variance. And you can see from the table, the first one was land use, urbanization, and the local watershed. And then different local habitat variables focused primarily on sediment type. A similar study was done by SQUIRP, and this is using all of our data, and they used a relative risk approach. Yeah, they used 24 different factors and basically looking at the risk of exceeding a threshold for CSCI scores based on exceeding a threshold for the environmental variables. And it broke down into habitat variables, water quality, metals, eutrophic conditions, eutrophic indicators, and nutrients. So here are the results. I try to color code them to make it more interesting and simple to look at. On the left, you can see that the 4 top variables that were related to elevated risk of having impaired CSCI scores were habitat variables and then eutrophic indicators or nutrients. So ash-free dry mass was the most important one, which represents algae growth, but also includes organic matter such as riparian detritus. The second most important factor was the IPI, which we haven't talked about yet, but that's the indicator for, oh God, physical habitat integrity. Was that right? Okay. It's like our CSCI score. It takes in a bunch of different metrics for physical habitat, scores from 0 to 1. So yeah, we see that habitat and nutrients and eutrophic conditions are important. And on the right, you can see all of the 24 variables in ranking order. And you can see that, for example, metals aren't as important. Okay. So kind of on a separate topic, now I'm going to talk about site variability. So in our region, many of the sites have been sampled more than once. Oh, 1 minute. Okay, cool. Basically, there's a lot of variability with, okay. Yeah. Okay, I got it. It's cool. So for example, where there are sites that have 3 or more samples, we have an average of 0.18 range for a CSCI score. And an average for all of our sites of about 0.22. You might think, that's not that much. But if you look at the thresholds for different classes, categories of impairment or condition, you can see that 0.2 could actually move you into a bunch of different categories. The plot on the right shows one of our reference perennial streams, and you can see the variability across time. Now, this is a reference site, so that variability isn't related primarily to any human changes. It's changes probably in rainfall or whatnot. And so if you were looking to assess this stream and you looked at 2010 or 2021, you go, it's impaired, but you're not really getting the full picture. So, okay, I'll get to my main points. So our recommendations is we need to understand our intermittent streams a little bit more, figure out a proper threshold. We also need to look at our urbanized channel streams and set biological expectations that are realistic. We need to look at local habitat variables and use similar analyses to assess what sort of factors are driving our biological communities. And again, consider interannual variation when assessing the status of streams. Sometimes using 1 or 2 might not be enough. And then we're gonna do a bunch of stuff in the future because I'm out of time. Yeah, okay, cool.
Thank you. All right, we have about 3 to 4 minutes for questions. So just raise your hand and I'll bring the mic to you.
Or you don't have to have any. That's okay.
This is the fun part, Christina.
I was just going to ask on the soft mixed channels, that you were looking at channel engineering. Did you look at those sites that scored really high? And
We haven't yet. So that's one thing. Well, one of the part of the thing is we don't have any more money to collect more bioassessments. So now my time is going to be spent trying to actually understand all of our data. And I think going to look at these sites that you expect to be scoring low but aren't, we should try to figure out why are these scoring so high? How can we protect them? How can we model this for other urban streams with limited ability to restore them? Yeah.
Any other questions? Do we have any questions online?
Sweet. So good. All right. Thank you all.
Thank you again. Next, we have Dr. Ray McNeish. She is at CSUB. She's an associate professor of biology and a freshwater ecologist with 16 years of research and mentoring experience. I like that. She earned her BS, MS, and MS from Millersville University in Pennsylvania. BS. And she also earned her PhD from the University of Dayton, Ohio. So let's give Dr. Ray McNeish a warm welcome. Thank you.
Oh, there we go. There we go. All right. Hi, everyone. I'm Ray McNish from Cal State Bakersfield. Today, I'm going to talk about the Kern River because it is in crisis. What you're looking at here is a video I took last August, 2024, in a refugium pool in the lower Kern River. And there are hundreds of mosquito fish and other stranded fish in this refugium pool. And in other parts, you find piles of dead fish that had happened during a rapid dewatering event we did not know was going to happen. And so during this rapid dewatering event, we documented over 3,000 stranded and dead fish over in 1.5 river miles. And so I'm going to share some information, a little bit of storytelling associated with this. And so what you're looking at here is one of our most downstream site. If you're familiar with Bakersfield at all, there's the Riverwalk Park. In the distance, there's Weir, the Bellevue Weir. And so this is August 23rd, Friday, 2024, and there is meaningful flow in the river. And then 7 days later, this is what that very same exact site location looks like because of the rapid dewatering event that had occurred. And so the rapid dewatering event is also linked to excessive water withdrawals, which, as we know, has a major impact on the ecology of freshwater systems. The Kern River is the 10th largest river in California. And it historically, like deep history, kind of seemed like maybe functioned ecologically as a perennial river, but it seems more to function these days like an intermittent ephemeral river instead because of anthropogenic activities. It begins in the Sierras, and water sources are stormwater, snowmelt, and we have dam releases as Isabella, that's upriver. Portions are classified as wild and scenic. It goes through several beautiful parks in the Sierras before it makes its ways here to the Central Valley. And it's very important for groundwater recharge in our area. And so zooming in, okay, so we have in the upper right there, the Isabella where the dam is, and then from there all the way through Bakersfield, this part is called the Lower Kern River. So it goes through a canyon and then lands in the valley and then piddles out west of Bakersfield. It doesn't typically reach the lake beds that it used to many years ago. There are a suite of diverse life that rely and interact with the river, including multiple endangered and protected species, okay, such as the Kern River golden trout. Historical uses, there were, of course, indigenous tribes that heavily relied on the river for spiritual and livelihood. More modern, like past 100-ish years, have been for agriculture, municipal use, and of course, recreation. And then we do have an unhoused transient community that heavily relies on the river, especially during summer months, if water happens to be there. In 2022, the Lower Kern River was named by American Rivers to be the 7th most degraded river in the United States due to excessive water withdrawals. And so you can see a biker. So we have the Kern River Parkway bike path that goes along the river. And to the right of it is a very common site in Bakersfield of a dry river bed. And so part of bringing awareness, there's grassroots and environmental organizations that also launched the first ever Kern River Festival to bring awareness and education for the public. Citizens are always very surprised to learn that their river looks like this because of excessive water withdrawals and not it's a natural condition. And so circled in yellow is attorney Bill McKinnon. At this event, this gentleman literally got on his knees and was like, there's a lawsuit, please collect data. We should manage what we measure, and people are not studying the ecology of the Lower Kern River. And I was like, okay, we'll start during the summertime. So we moved up our plan instead of starting monitoring in the fall when my sabbatical started to doing it during the summertime because this gentleman literally, truly got on his knees. And this was a really good thing. So it's good to listen to people who make these requests because we didn't know the river was going to be rapidly dewatered before the fall started. Okay, and so this created an opportunity when the dewatering happened to understand how rapid water loss affects the ecology of the stream. Okay, and so some predictions that I think are logical that people in this room probably would have is that with the reduction of water availability, reduction of aquatic wildlife, we also expect there to be more anthropogenic litter. So side note, I study microplastics and trash a lot in the environment. And then of course, reduction of river habitat, and with a reduction of water, warmer water and less oxygen to support aquatic wildlife. Okay, and so here is satellite of Bakersfield. The blue are 5 out of the 7 weirs that are on the river, and right above these weirs are canals. And so a lot of times the weir gates will be closed to help divert water to the canal system so it's reused for other purposes. Okay, and so upstream of the first set of weirs, the Beardsley Weir is our, quote, reference site, Hart Park. It flows year round, and oftentimes the flow is pretty dangerous and you can't really go very far into the river, you'd be carried away. All right, and so downstream of 4 weirs, we have in-stream, in-river monitoring sites that we monitored weekly last summer, okay, and then monthly since then because, well, the river's been mainly dry. Okay, so our most downstream site is the first site that I showed you a picture of, the Bellevue Weir at our Riverwalk Park. All right, and so our monitoring field methods, okay, for weekly last year summer was to use YSI for physiochemical parameters, and we took triple cut measurements at each site, water sampling for chemical contaminants and nutrients, water depth, all right, and then we don't have scientific collector's permits. We are working on getting, I'm an aquatic macroinvertebrate entomologist. So fish, I mean, I study fish, but usually it's like how much plastic they eat, okay? And so learning the fish, okay, please talk to me about that. But this is mainly just like observations. We do have eDNA for fish and also algal communities as well, okay? So our sampling timeline, so like the story I'm going to share is mainly pictures, some data, okay? So we had massive water presence in the river system because of the 2022-2023 rains and snowmelt. And so there was continuous flow in the Lower Kern River for about 18 months to the point where excess water could be diverted to basically fill up all the groundwater recharge basins. And Kern Water Bank is one of the bigger ones. It's the first time they were able to do that in 7 years. Okay, and so we did weekly monitoring during summer 2024, and then surprise, dewatering event happened. We didn't know it was going to happen until we showed up, and there was like basically no water at our sites. And then we've continued to do monthly monitoring, and then add some other monitoring at other parts of the river that get like wet and dry and wet and dry. Okay. So data collection is underway, samples are still being processed. All right, so a couple of data slides here. So this is the data from last year's summer where we're doing our weekly monitoring. And so the sites are color coded. Green is Hart Park, the reference site, most upstream. And then the lines and the colors get warmer as we go downstream, with red is the most downstream site. Okay, so unsurprisingly, water depth significantly decreases as you go from upstream to downstream. Not surprising, since we've got those weirs and water diversions are happening. Okay, if we focus here at this site, that's the one I've been showing you pictures of. So on the 23rd Friday of August, this is what it looked like. And then literally 7 days later, it looked just like this. Okay, and again, we didn't know this was going to happen. In terms of temperature, okay, and so here's our temperature across sites through time. And we did see a significant temperature effect. And I drew these lines on here. Again, I'm not a fish person, people, okay? So help me out if you can. But I had looked up on the EPA website, and 19 to 24, this is really important in terms of, temperatures for cooler water and warmer water fish and prolonged exposure. So you can see we definitely have temperatures that are, maybe above that high health risk that I saw on the EPA website. A refocus here, which again is our most downstream site at the Bellevue Weir. I also did have temperature loggers put in at some of these sites that were measuring temperatures throughout the day. Okay, and you can see that it's really warm temperatures. Okay, and there are, there's warm spots and cool spots on the river, but there's also what we started calling jacuzzi spots. You literally can feel yourself sweating while you're in parts of the river, and it's like hip deep. Okay. So this is, concerning for maybe, aquatic wildlife through prolonged exposure. And this is like over 7 days in our high and a low, okay, for our ideal pattern. And so here in terms of dissolved oxygen, it's percent. And so if I put a line there, that's 100%. We expected, warm water, lower oxygen, but how are we getting this 100%? There actually was a very healthy aquatic invasive species populations all throughout. And so we can literally see the oxygen values on the YSI probe increase the closer we got to like the flotilla of the water hyacinth. And then of course there's rooted vegetation as well. Some parts of it do look like an aquatic underwater forest. So this is adding probably a lot more oxygen to the system, even though we have these really warm temperatures. If we average oxygen overall, we do see that as we go from upstream to downstream, there is a significant reduction in oxygen in the water to support wildlife. When the river was flowing, we had your typical Riffle 1 habitats, just as you would expect. For reference, some of these pools are 5 feet to 15 feet deep, and so we do snorkel in them. And we have seen some of our colder water fish. So for example, we have seen rainbow trout in some of these pools. Aquatic macroinvertebrates, we didn't have the permits, but we have observations. So, we're working on the permits. Hopefully we'll have that in a couple weeks. But our, clams, we got a diversity of dragonflies and damselflies, as well as mayflies and caddisflies. We haven't observed any stoneflies in this reach. And then here's just some underwater photos of, bass and other fish that we got while we were snorkeling. So that was our observations in terms of our weekly monitoring. And then the dewatering event happened rapidly through multiple miles of the Lower Kern River that runs through Bakersfield. And so as this was happening, we were going out and revisiting our sites much more frequently, like every couple days. All right. And so we lost our riffle habitats and basically became more stagnant, marsh-like, and much more common were refugio pools, especially those deeper pools. Okay, and you go out and this is our Stockdale most downstream site at the Bellevue Weir. And we actually, if anyone knows Lois Henry, reporter from SJV Water, she was with us just to do a story. And so she saw our raw reactions when we showed up and the water was basically gone. And there were over 300 dead and stranded fish. In that pool right there, that's about 14 centimeters deep, and it had oxygen that I think, if I remember correctly, was less than 4 ppm. And there were over 90 carp still alive, basically shoulder to shoulder crowded. All right, and so upstream is what we call the designated Bakersfield Environmental Studies area. Okay, that whole brown floodplain there. Okay, and the river, and the University of Cal State Bakersfield is literally just like south of that, across Stockdale. Okay, so this is just north of our university. This is about 1.5 river miles. Okay, and so we do what's now the water, when water was still here before this part was dewatered. This is upriver from our downstream site, most downstream site. We did a fish survey. So we went out and we surveyed for fish. This is field observations because we don't have permits. Okay, so if you're really interested in this, we have a whole poster. My student Stephanie will present on it, fish in different other sites too, but just some glimpses, teaser trailers. In yellow was the most common that we saw during this water fish survey. So you have carp and bluegill, sunfish, and bass, for example, were the most common that we saw. After the dewatering event, as you expected, it's a dry riverbed, except for maybe some really deep refugia pools left. Okay, and we also saw a major uptick in anthropogenic litter or human trash. I should have been primed to study that because I study that in the environment, but what we think was happening is as the waters were receding, fish can't go upriver because there's weirs in the way and there's no fish passage, but humans can relocate upriver. So they were abandoning the items they collected, go upriver where the water is, they collect new items, abandon, and so we think this is what was happening, and we are studying this now in the river. And so we, a week later, the BESA, the Bakersfield Environmental Studies area, was like this. It was dry. It went from being a full flooded, flowing river with pools 5 to 15 feet deep and to almost basically no water. And so we did our transect survey again, and there were piles and piles of concentrated dead fish, stranded fish, and we took hundreds of photos. My student has done over 2,000 measurements in Image J. Please see her poster, okay, and helping, give her attention. All right, and so these are examples of photos, and she's been measuring their size and whatnot. So we, fish can't swim and hide from you when they're dead, so you can, of course, your tax awareness increases, okay? And so, for example, the Sacramento sucker, we did not observe that before in our survey, and then, of course, some other taxa as well. The eDNA actually aligned nearly beautifully, perfectly with our field observations, which made me feel good as not a fish expert, okay? But new taxa was the threadfin shad. We didn't see that in our observations, but that got picked up in the eDNA. What did not get picked up is the rainbow trout, and I'm not surprised because it was probably really low populations and maybe not enough eDNA in the water to pick that up. But we have seen them in different stages, juveniles to adults in different parts of the Lower Kern River. So overall, to kind of summarize what I've been sharing with you, not too much of a surprise here. Water reduction, we saw an increase in trash, generally an increase in water temperature, reduction in oxygen, and exturbation of all aquatic life. And so this actually led to over 30 news article releases, including the front page of the LA Times last fall. And then I wrote a public letter that I read at a city council meeting and to notify the council and the people of the city of Bakersfield, but then also to encourage that as these weirs are being rebuilt and fixed and whatnot, please include fish passage so at least some of them can migrate upriver when you're doing an intentional dewatering event or you're going to divert excess water. And so we are continuing to study the river and work with these community groups of interest. There is a lawsuit in play and my data is being considered for that lawsuit. So I've been making parts of it publicly available. So all interested parties could use it if they want to. There's also river restoration activity planning that's been happening. If anyone knows the entity organization River Partners, they have been working on a riparian restoration, and we've been in discussions with them. And so I know I'm in my question time, but sorry, new thing happened. The water came back to the Bakersfield Environmental Studies area Friday. So you can see mirage of water there, okay? This was Friday at noon. This is 8 o'clock the next day, 8 a.m. And so we saw it at noon and my team, we went out and then we tried to do a fish transect survey. We didn't see any fish, but we saw one giant invasive crayfish, okay? And I'm sure there are fish there, okay? And all of the riffle run habitats, all the deep pools are filled with water. And it might be hard to see, but we saw hundreds of dragonflies that were in tandem and laying eggs. And so while we didn't see aquatic You know, macroinvertebrates, the adults were there in less than 24 hours laying eggs, which is pretty cool. So thanks to all my students and the collaborators and people who have been helping out with this research. And we'd like to acknowledge the original, not settlers, oh my gosh, the original indigenous people who are living here, and we continue to promise to be good stewards of the land. Thank you for your time. I maybe have time for a question.
Thank you, Dr. McNeish. Again, if you have questions, I'll bring the mic to you.
Thank you. Very, very sobering story you're telling us here. With regard to the oxygen concentrations, you take that just during the day. I know you're talking about the invasive plants that are putting a lot of oxygen, but at night, when they're respiring, it may really plummet the oxygen concentration. So
this is a great question. So yes, the oxygen was measured in the day, and we would go out first thing because it's hot. So it's like 6:30 a.m. is when we're out there. To be honest, it didn't occur to me to measure the oxygen at night, but we should be doing, you're right, like that's something that we should consider be doing. Yeah.
I was just wondering, in the management plan for the river, is that a common thing for the dewatering?
Is dewatering of the river common? Is that what you're asking?
Well, yeah, I mean, is there a requirement to have a certain amount of flow? Like a lot of the rivers like have a certain CFS, whatever.
So this is what the lawsuit is about, is that Fishing Game Code 5937, generally operators and owners of dams should have water downstream for fish to be in good condition. And also the public trust, California Public Trust Doctrine is another aspect of the argument. And so it's a bit more complex. I'm not an expert, but we can talk more about it later if you want to. But basically, no, there is not a requirement. And so there are multiple entities that are, the water is diverted to support that goes to different water entities throughout different times of the year. Some of it goes to the city of Bakersfield, which is fighting to get water in the river year round also, but they are actually named in the lawsuit, so. Yeah, more like water for human use, not really for the supporting a river.
Hello. Just for curiosity's sake, when you were collecting your eDNA samples, I'm assuming from water samples, right? Were you collecting them at different points in the hydro period, or did you just do like one, collection and then run the test?
So this happened a couple weeks before the dewatering event, and at the time I wasn't prepared for the type of data the attorneys were hoping for. And the kits, we did these kits because they needed data super fast for their court hearing, which ended up being delayed anyways. So it was one eDNA sample kit at each of our 5 sites. And so they paid for those, but since then I have been buying more and we've been doing more frequently in terms of that, but it's hard when there isn't water in most of the river most of the time. So hopefully we do replications and we can do seasonal. This is the goal. And so I'm just stockpiling the kits at this point. Yeah.
All right, so that's time for questions. Thank you again, Dr. McNeish, for that wonderful presentation. Next up, we have Dr. Rafael Mazor. He has a PhD in environmental science, policy, and management from UC Berkeley. He is a freshwater biologist at the Southern California Coastal Water Research Project, where he leads the bioassessment research.
Thank you.
Yes. 5 minutes for time, yeah.
Hey, great. Can everyone hear me okay? How about now? Yep. Okay, great. So thank you for having me. Rafael Mazor from the Southern California Coastal Water Research Project. I am, again, coming here to talk to you guys about modified channels. So it's a topic that has become near and dear to my heart. You heard Christina touch on the subject a bit, so it's actually a really great setup for what I'm going to share with you today. And so I'm sharing the results of a study that the Southern California Stormwater Monitoring Coalition recently concluded. And so I've got some good results to share with you. So many of you understand that modified channels, they experience a lot of different kinds of stress, whether it's water quality stress or altered flows or lack of habitat. And some of these stresses are direct consequences of channelization, things like the loss of habitat. Channelization can also exacerbate the impacts of other stressors, particularly things like heat stress and water quality. So, there can be a really profound impact on biological communities. In Southern California, we've been working with a classification system for modified channels that really focuses on bed and bank material, similar to what Christina presented in the Bay Area. We have streams that, you've hardened the stream bed and the banks, typically with concrete or riprap, but you also have a number of soft bottom channels that have a bit more diversity in what we've done to them. We might have hardened one or both sides or none at all, but these are all different kinds of engineered channels. And at this point, we have a pretty clear idea of what the biological conditions are in these different channel types. The data I'm showing you here is actually statewide data, but primarily driven by data in Southern California. And we know that hard bottom channels, it's pretty rare to get your bioassessment index scores close to reference, above that 0.79 threshold that Christina and Chad had mentioned. It rarely happens. Soft bottom channels, it depends on the channel type. When you're talking about the one-sided channels, it's actually quite common. These are pretty unusual for engineered channels. These are typically occurring high up in the mountains where you're putting in riprap to protect a road or some property, but there's little alteration on the other side of the valley. The other kinds of soft bottom channels are more pervasively altered. And in those cases, high scores are less common. More common than you see in a hard bottom channel, but still fairly uncommon. So, okay, we've documented that these streams have a lot of problems. Can we start finding solutions? Can we do something to improve biological integrity? So to get at this, the SMC had a study that looked at sort of a few different ways of examining this question. First, we wanted to know what's the relationship between water quality or habitat stressors and index scores in these different modified channel types. So that could give us a hint if reducing stress there could improve conditions. We also wanted to look at flow. This is one of the tools at our disposal for affecting condition, as you just heard from Ray, that flow has a profound impact on biological conditions. So can you set flow targets for these types of streams? And then finally, we wanted to know, can you identify streams where if you put in these sort of practices to change conditions, but leave the modification in place, is that likely to benefit, result in like an improved CSCI score? So I'm going to share you sort of the results of these questions today. So this slide shows you a typical relationship that we see looking, it's over 1,000 data points where you have CSCI score on the y-axis and on the x-axis. That's a measure of water quality condition. In this case, I'm looking at specific conductivity. This can be like a direct measure of stress itself, but it's also a really great indicator of other water quality parameters that may have been introduced into the water and elevated conductivity, but you haven't measured those directly. And what you see is that blue line there, those are natural channels. And you see it's a fairly steep negative relationship. And this is kind of what you'd expect. That green line, those are those soft bottom one-sided channels I mentioned earlier. And it's pretty much the same relationship. You have really high scores when the water quality has very low conductivity, and they kind of decline rapidly as that stress increases. Now you see a somewhat different story when you start looking at the other classes of modified channels. Those relationships get quite a bit weaker. They're sort of shallower. They intercept the y-axis sort of lower, which sort of suggests that even if you brought that water quality all the way to the left, things would still not be as good as you get in a natural channel. And when you look at the hard bottom channels, it's almost pretty much a flat line. Now, there's a couple different ways to interpret this. You could say that these types of channels, you're not going to get much bang for your buck if you fix the water quality. But there's actually a lot of problems. There's a lot of things going on in this at these sites that we might not be capturing in these models. A lot of these modified channels occur in areas that receive a lot of different kinds of, urban or agricultural runoff. So there's a lot of stresses that we might not be capturing in this model, and that might explain what's going on as well. Now, when we looked at a slightly larger range of water quality or habitat stressors, we kind of see the same thing. The natural channels and those one-sided channels, you tend to see a fairly good response. Those engineered channels, it's muted. Maybe you might not have had enough data to really meaningfully look at that relationship, but when you did, it tended to be flatter. Now, again, all these panels are showing you that we're looking at one stressor at a time, so it's still
sort of a limitation of these analyses, and I'll touch on that a bit more later. We also looked at the effective landscape, and this is actually kind of like inspired by some of the studies that the Bay Area done in the study that Christina mentioned. And when you look at sort of two different measures of channel alteration, either the percent development in the landscape or road density, you do see that there's sort of a muted response in the engineered channels, but there's actually pretty good evidence that at these less developed watersheds, conditions can be better even in these modified channels. And I'm highlighting there an example of one of the sort of rare exceptions where it's a hard bottom channel with a very high index score, comparable to all of our reference sites. And you can see that it's pretty odd. It's almost no development in that watershed. So we understand that there may be a case to be made that the modifications themselves are less important than all the other things going on in the watersheds. So this is something we're investigating further. So I mentioned we're looking at flow as one of the possible things you can manage to improve or protect biological condition. And we have this, we've been building this great toolkit. You'll hear actually more about this, tomorrow. But we have, a toolkit that helps identify ranges of flow conditions that are associated with good biological integrity. Now, most of these flow ecology models that we've developed are really geared around reference conditions. That is, can you get your CSCI score above 0.79? And as I just mentioned to you, that's not always a very easily achieved goal for our modified channels. And so these tools tend to be less useful there. So we decided, can we adapt them to make them more flexible and more meaningful for some of the goals you might want to set for these modified channels? The goals we're looking at are basically alternative thresholds based on not the lowest of the reference sites, but the highest of the modified channel sites. Can you get a score up to like the best that those channels can do? And so we looked at these sort of best observed thresholds for 3 different types of engineered channels: the soft bottom with no hardened sides, the soft bottom with 2 hardened sides, and the hard bottom channels. And you can see that the numbers are a little bit lower, quite a bit lower for the hard bottom channels. And this graph sort of shows you this general approach. This is a model showing how CSCI score responds to changes in a flow metric, in this case, summer base flow. And the way you look at this is that you can actually have stress increasing in both directions. On the left side, these are depleted flows, similar to what Rave's showing us. And on the right side, you have augmented flows, and you have stress in both directions. And in the middle there is where you see these, what we call natural flows, and that's where we have that 0 on that x-axis. So you can look at a site, and that's sort of represented by that blue dot. You know what its CSCI score is, and you can know how much its flow has been altered. And you can figure out how much work would it take to achieve one of those different targets for biological integrity, whether it's reference condition at RC. The table got a little bit reformatted there, I apologize. Reference condition, the best observed threshold, or what we're calling an incremental threshold, where you're kind of not trying to set a target, but rather just an improvement in that CSCI score by, in this case, a value of plus 0.1. So in this case, you're going from 0.6 to 0.7. How much work would it take to get there? So you can take like a lot of improvement in flow to reach reference condition, slightly smaller to reach that best observed condition, and smaller still to get that incremental improvement in CSCI scores. We then asked, okay, if you do a modest change in flow, which we kind of arbitrarily defined as a 10% improvement in flow, which goal would you achieve, if any of them? And of course, there could be cases where you have a low CSCI score, but the flow itself hasn't been altered, so it's other types of stressors. So these are sort of how we classified our sites. Now, perhaps surprisingly, we discovered that flow wasn't always a problem. A lot of the sites that we had in poor conditions actually had flows that were close to that natural range. And that's that sort of green part of that stacked bar. That's the non-flow related. When it was a problem, it tended to be a major problem. Large changes in flow would be required to get some kind of improvement. Those red bars show that even with like sort of greater than 10% improvement in flow, the CSCI would not increase that much. When you look across the board, across all these different flow metrics that we considered, the general story was that you could get incremental changes with moderate improvements in flow. So that's our overall conclusion here. So what about all the other stressors? Is there going to be a meaningful benefit to improving other stressors? And the way we answered this question is looking at our causal assessment tools. Specifically, we have a toolkit that we call Rapid Screening Causal Assessment, or RSCA. It's based on some methods that the EPA developed, the Causal Assessment Decision Diagnosis Information System, but sort of streamlined and adapted to sort of the data sets we have in California. Now, one thing to know about this causal assessment is that it relies on comparator sites. Comparator sites are sites that have similar environmental conditions to the test site you're trying to evaluate, but have different levels of stress and presumably different levels of biological condition. So in this example, that green dot at the bottom in San Diego, that's our test site. And all the yellow sites were deemed to be similar comparator sites to that site. And most of them are also in SoCal, a handful in Northern California. And the black dots are sites that were environmentally different, so we don't use those in our analyses. But we still have a pretty large data set to work with. It's several hundred sites. But when we're talking about modified channels, we thought, what if we just look at the subset of comparator sites that have the same types of modifications? So you're comparing our hard bottom test site to other hard bottom comparator sites. And I just want to point out that the vast majority of those sites are in Southern California, largely because we don't have a lot of data on channel engineering outside of SoCal. So the way we derive evidence, this is just an example. One line of evidence is called spatial co-occurrence. The idea is that you're trying to ask if stress levels are lower at healthier comparators. So it's not all comparators, just those with a higher CSCI score. In this case, you're saying your test site might have lower total nitrogen than your healthier comparators. That means it's not likely a problem. Total nitrogen is probably not contributing to poor CSCI scores. If it's somewhere in the middle of that range, you would say it's indeterminate. And if it's well above that range, then you'd say this is strengthening evidence. So that's evidence that total nitrogen is the problem and reduction could improve things. But that's our traditional analysis. If you pair it with this modified analysis, you might get a different conclusion. And in this case, we're finding that while it's higher than healthier comparators, it's right in the middle of the range of healthier, similarly modified comparators. So while total nitrogen is probably a contributing factor, there are other things going on at this site that are relative to healthier modified channels that are affecting it. Now, we often found that this modified approach confirmed our conclusions. And here's an example from Conejo Creek in Ventura County, where we saw that total phosphorus was substantially higher than healthier comparators, whether you looked at the full data set or just the similarly modified comparators. And this is the case of about half the time for when we did the two analyses in parallel. But sometimes the other half of the time we saw different conclusions. And typically we saw it move like in this pattern where your traditional analysis showed that it was a likely stressor, but you were right in the middle of the range of the modified comparators. So this is the case of the San Jacinto River with elevated conductivity. It wasn't elevated beyond the state of healthier, similarly modified channels. Now, we also had a problem with data deficiency with this modified approach. Now, here's an example of Murphy Canyon in San Diego. This is actually right next to the water board office. And even though it kind of looks pretty lousy, it scored not that bad, 0.71. This is among the healthiest hard bottom channels we have in the region. So that meant that we didn't really have healthier comparators to look at. So we really couldn't do this sort of modified approach here. So overall, across these sort of three lines of evidence, we saw that reducing stress and or managing flows, there's some potential for improving conditions, but that potential varies from site to site. And there may be fairly minimal benefits at some sites. And therefore, we think this needs to be sort of considered when you're trying to prioritize sites for which ones you're going to do an intervention at or what stressors you're going to try to fix. Now, we do have a new project that's getting started that sort of builds on the findings we've had so far. First, we're going to be looking at revisiting those models I showed you earlier of how water quality affects CSCI scores. But we're trying to look at cumulative stressors at once, looking at this approach proposed by Feld et al. We're going to look at the impact of channel maintenance to see if there's certain strategies or timings that might have more or less impacts on CSCI scores. And finally, we want to look at the impact of restoration. So these are often, looking at places where we've removed the channels. And I look forward to picking Caitlin's brain about some of these restorations in the future, and Ray as well. And just last, I want to just add a little plea for data. We don't have a lot of data outside of SoCal. We have this dashboard that you can, scan that QR code or go to that website and look at how we've classified all the sites we have in the state. You can either say, yeah, you got that right, or you made some mistake, and here's the correct classification. Or we might not have a classification. Those gray dots are all the sites where you're lacking data. You can tell us what you think it is. And that would really help us expand these analyses in the future. And with that, take question. Thank you.
Thank you so much, Dr. Mazur. Any questions?
Based on what we just heard in the Kern River of the dewatering, have you got an idea of the flow regimes that are going on there as a real stressor?
You know, I can't really say much specifically about the Kern River. I imagine that you said it was historically a perennial river, so clearly that situation that you documented is not within normal historical range. But yeah, presumably flow management could be a major part of that restoration strategy there.
Thank you. Hey, Rafi, great presentation. I really liked it. I'm kind of curious, is the end result of this work going to be kind of like the best that you can be if you're a modified channel, like conditions describing the best that a modified channel can be, the healthiest that can be, sort of different characteristics? Or is the end result effort or things that managers can do to improve their modified channels? Or is it a kind of a combination of both?
Well, I would say that you're really hitting on, like, you know, one of the key areas of debate that folks are having, particularly between regulators and regulated parties, like what's the best these channels can do. So what we're trying to do is, you know, help folks like at least sort of understand on a common page where we think that, you know, some kind of interventions are likely to pay off, and so maybe they can agree to prioritize those sites. But yeah, there is sort of a big debate about what standard to hold these modified channels to.
Yeah, so it sounds like you're sort of looking at it from two different perspectives as well. So you're also going to give managers tools that will help them to manage their modified channels.
Right, right. And the recognition is that the modifications are likely to remain in place for many of these channels for the foreseeable future. So what can they do if that's going to remain in place?
Hey, Rafi. Thanks. Great talk. One thing I feel like that we haven't really captured in our swamp bioassessment is documenting modified channels and how close they are to natural conditions upstream.
Yes.
I think both of your example sites are within 100 meters downstream of natural conditions. So I think it would make our data more robust if we develop some means of capturing that.
Yeah, so I think what you're saying is that when you have a modified channel that tends to score well, you're often going to see those closer to natural areas, even if they're not in a natural area itself. And yeah, you're definitely right. We have a few analyses that kind of back it up. I mean, I'm curious if you think there's a good field protocol. We ought to, some addition we should make to our field methods for that, rather than relying on some of the GIS analyses. But I think that's going to be a key explanatory factor.
Thank you.
This is a comment from the chat from, I suspect, someone you know. Great talk. Go Bears.
Okay.
One more question. We have time for one more.
Thanks, Rafi. So when we're looking at modified channels and doing comparisons for healthier, like for example, you had a total nutrient slide showing hardened. Did you consider using actual water quality criteria as a backstop? So for example, like in your hardened streams, like we have in Region 9, we have a TN objective of 1.
Mm-hmm.
And so pretty much all of them were above the 1.
Yes. Yeah.
So it's like, okay, healthier, quote unquote, but if the water quality is all still crap, for lack of a better word, how can you incorporate that in that evaluation process?
Well, I mean, we're defining the healthier streams strictly based on CSCI scores, because those sites all had, you know, higher scores. It might be just a little bit higher. It might not be a meaningful difference. So I guess I'm not really clear on how we could use the water quality data other than to sort of evaluate differences between those two classes of like the test site and the healthier sites.
Yeah, I would just suggest not losing that resolution as a potential stressor in the broad scheme of things.
So like the conclusion in those cases may be sort of less reliable. Yeah, that's probably the case.
Thank you, Rafi. Next, we have Holly Swan. She is a regional manager for California Trout, a nonprofit organization working to revitalize waters for resilient wild fish and a better California. Holly has a master's degree in environmental planning and resource management from Chico State, and she will be presenting on the Iron Canyon Fish Passage Project.
Yes.
So let's welcome Holly.
I also have my colleagues here, Madison and Michael from Fish Bio. So we're going to do like a little round robin presentation to keep you guys on your toes. Okay, so yeah, we're going to present on the Iron Canyon Fish Passage Project, an ecosystem-based approach to restoration. We'll provide you with some restoration updates and an overview of pre-restoration monitoring. We'll provide a little bit of background on the watershed, Big Chico Creek, talk about the Fish Passage Project, and then some pre-restoration monitoring, and then just give you a little like outlook overview of future opportunities. So Big Chico Creek is located in the Sacramento River, it's a tributary to the Sacramento River, and it's located between Mill and Deer Creek to the north and Butte Creek to the south. This is very important because Big Chico Creek does get straying spring run Chinook salmon, and it's about 45 miles long. It has a natural hydrograph, which is pretty rare in the Central Valley, as you most know, and it has a varied land use. I think the most important thing to mention on the land use is many miles of the creek run through a nearly 8,000 acre ecological reserve, the Big Chico Creek Ecological Reserve, and a nearly 4,000 acre municipal park called Bidwell Park. And just to note, there is the orange arrow is where Iron Canyon is, which is located in Bidwell Park. This is a graph showing the average daily flow in cubic feet per second on Big Chico Creek over the last 12 years. This is here to really show you the dynamicness of this creek, especially in Iron Canyon. So the photo on the left is the Iron Canyon project reach at base flow, which is about 30 to 35 CFS. And then the image on the right is about 5,000 CFS, same reach of the creek. Just this last year, it got up to 10,000 CFS. So as you can imagine, anything we do here for fish passage has to be able to withstand some pretty intense hydrological forces and also be robust to sediment transport. So Big Chico Creek historically did support steelhead and spring run Chinook salmon, and it also had a diverse array of other native fish species. A little history here is that there was a rockfall. No one really knows when or why, but a rockfall did create a fish passage barrier in Iron Canyon. And to assist salmonids to get through the canyon in 1958, CDFW did install a 17 concrete fish ladder weirs, which have become dysfunctional over time, and they no longer are able to pass salmonids. And then in the 80s, due to a sort of perceived notion that other native fish like pike minnow and hardhead were out-competing salmonid fish, we wrote and owned the creek between Iron Canyon and Higgins Hole, which is the furthest extent of anadromy. So 8.5 miles were poisoned in the 80s. Iron Canyon is the only total fish passage barrier in Big Chico Creek. Again, there's 8.5 miles of cold water holding and spawning habitat above Iron Canyon, but right now, any straying spring run Chinook salmon that end up in the watershed are stuck in the aptly named salmon hole, which is the image on the left there. And the recreational use is too high, the water temperature is too warm, and any spring run Chinook that do stray into Big Chico Creek often die before they are able to spawn. So the Iron Canyon Fish Passage Project, the approach for that is we are using a stream simulation design approach, meaning we're taking a reference reach just downstream of Iron Canyon, and we're looking at the fish passage conditions there, and then we're going to emulate those same hydraulics in the project reach. And to do that, we're going to remove all 17 of those concrete rebar fish ladder weirs that no longer work. And then we're going to create a series of steps and pools out of the basalt rocks that you see in the photo there. And the end result will be a passable natural fishway at a wide range of flows that will be passable to salmonids as well as other native non-salmonid fish. And if all goes as planned, this will be completed by the end of 2027. I'm going to pass it over to Michael here.
All right, thank you, Holly. Yes, this goes to speak to the ecosystem-based approach of this fish passage restoration. So we do, while there are salmonids in the creek and we do care about the salmonids in the creek and the funding came based on salmonids being in the creek, we very much care about fish species and other animals beyond salmonids. So this is the native species assemblage here that we see in Big Chico Creek, starting with hardhead on the top left, and then of course the rainbow trout, sculpin, spring run chinook salmon, sucker, lamprey, pike minnow, as well as California roach. What I do want to point out about those fish species is, and Maddie's going to speak a little bit to that here in just a minute, we do have some longer running pre-existing baseline data to evaluate fish species community composition as well as distribution in this creek. But not all these species are readily observed. So usually we conduct snorkel surveys to do abundance estimates and distributional surveys. But some of these species, like the juvenile lampreys that live buried in the soft substrate as amicetes, really difficult to detect while snorkeling, for example, or the sculpin, which are small cryptic, typically live between in the gravel and between the cobble. Very, very difficult to count as one is snorkeling. So one of our monitoring approaches to this baseline monitoring that's currently ongoing is also environmental DNA. Also, maybe just to briefly mention, one of the components of this project is to not only facilitate volitional passage of these native species back into reaches they historically inhabited, but also to kind of jumpstart that process by reintroducing some of these extirpated non-game fishes above that Iron Canyon fish passage barrier. That's already been occurring opportunistically from some, and I don't know if you can recall the map, the watershed map showing earlier, there's some tributaries to Big Chico Creek that enter Big Chico Creek fairly low down, Mud Creek, Rock Creek. Some of those are intermittent and do dry up in late spring, early summer. We've been conducting some fish rescues and I think so far have relocated 1,000 plus or so individuals belonging to these different species upstream of that barrier that's about to be remediated here. And with that, I'll pass it on to my colleague, Maddie.
Thanks, Michael. Michael dipped into this a little bit, but these slides show you our pre-restoration monitoring efforts relating to fish and beyond. He mentioned the snorkel surveys, so we're looking at fish distribution and abundance in the creek. And fish bio, we love Big Chico Creek, flows through our backyard. We've had the opportunity to conduct annual surveys in collaboration with CSU Chico and also Big Chico Creek Ecological Reserve. So dating back to 2013, so we have access to a large amount of data that will feed into our baseline snorkel data. We're also doing eDNA. So we're looking at the fish community in the creek and then also beyond fish. We're able to detect mussels, amphibians, turtles, etc. And then of course we're doing BMI monitoring under the SWAP methods, of course, doing reach-wide benthos, BMI collection, and also the PHAB survey. That's been going on for about 3 years now, and we do annual sampling for all of these pre-restoration monitoring efforts. And since the large goal of this project is to restore connectivity for salmon in the creek, we are interested in doing pre-restoration monitoring, looking at environmental parameters. When it comes to temperature, salmon require a very specific threshold to survive as juveniles. they need specific temperatures for rearing, and then as adults for oversummering and holding in cold water pools. And so to look at this thermal suitability and effectively suitable habitat for salmon in the creek, we've deployed temperature loggers at 9 locations, from Beeser that we've mentioned before all the way down to just upstream of the Sacramento River confluence. And these have been in operation since August of 2023. We're also looking at connectivity. This is another poorly quantified factor in Big Chico Creek. We know that in the summer months, the creek loses its surface flow and dries up in lower reaches, but we don't know for how long, really, when that occurs, where it occurs. And so we have deployed pressure loggers, and we are monitoring water level and seeing if we can identify some drying patterns with which we can inform future stranding, potential fish rescue surveys as salmon come back to the creek. Lindo Channel is a flood control bypass channel where high flows are diverted out of Big Chico Creek in the winter months, and so that presents a potential alternative migration route for salmon, and which introduces the risk for stranding. So yeah, these are a few of the environmental parameters that we're looking into. And now we're going to hand it back to Michael.
Yeah, we'll do a couple more switches back and forth here. This is just a very brief overview of that baseline, in this case, rainbow trout abundance survey that's been going on over the years. Perhaps three things to take away from this slide. First and foremost, natural population abundance fluctuates pretty dramatically. Second thing is they do tend to do a whole lot better, or we see higher juvenile abundance, higher reproductive outputs during wet years. So wet and above normal years typically result in a big jump in juvenile abundance the following year. And also, I know they're mentioning this the first time here in this presentation, but we did have a very destructive fire in the watershed just last year that burned a large proportion of this watershed. Was the Park Fire. We were able to conduct the snorkel survey shortly thereafter and actually estimated our highest abundance since. So if that fire did have an impact on the aquatic community, it was at least too early to reflect on the fish community at that point. We do have, I think, similarly promising indication from the BMI community, at least initially, that Maddie will highlight here. But yeah, all that to say, the immediately feared kind of scorched earth or scorched creek effects of that fairly catastrophic Park Fire have not manifested yet, at least not as far as the fish are concerned and not as far as the BMI community is concerned. But I'll let Maddie say a couple words about that.
Yeah, so this is just kind of a little sneak peek into some of our BMI results. If you were paying attention to Nathan and Chad's presentation earlier, the California Stream Condition Index, there are a lot of caveats with that. But yeah, just a little sneak preview of our 2 stations that we have been monitoring over the last 3 years. Salt Spring Meadows, which is at the upstreammost boundary in the creek on the Beeser Reserve. And then there's a little typo. Ford is actually not downstream of Iron Canyon, so just ignore that. That is just upstream of Iron Canyon in Bidwell, Upper Bidwell Park. But yeah, you can see that over the last 3 years, CSCI scores have been relatively consistent, with the exception of 2025 at Ford, has kind of jumped up into the possibly altered zone, which, yeah, could potentially indicate positive influence on the stream following wildfire. But yeah, again, we're in the pre-restoration monitoring phase with all of this still. And yeah, we're looking forward to
digging into some deeper analysis as we move into the post-monitoring phase of Iron Canyon. Some of the things that we're certainly curious about are, one, the relationship between like how does wildfire affect BMI, and then what is the relationship between fish and BMI, specifically salmon, who would be bringing more marine-derived nutrients into the upper creek ecosystem, and then yeah, just the relationship between BMI and other environmental variables that we are tracking.
All right. Just, yeah, very briefly, a little bit about next steps and conclusion. We've mentioned a few times this is intended to be a very, holistic and inclusive restoration project. So one of the things that Maddie just mentioned, we're definitely going to continue post-fire watershed monitoring and continuing that recovery, or monitoring that recovery post-fire, and then any potential trends or step changes that result from reestablishing connectivity. Definitely monitoring recolonization of salmonids. That's going to be probably one of the easier aspects of this. And also, beyond salmonids again, and beyond just fish, monitoring for changes in mussel abundance and distribution, and of course the BMI communities. The hope for this project is, of course, to contribute to the overall population. An evolutionary significant unit, ESU, is the management unit for these anadromous salmonids. So continue to contribute to the resilience of this listed ESU, or those two listed ESUs. It's both Central Valley steelhead as well as spring run Chinook salmon, and of course to serve as a model for nature-based fish passage. So the last thing anyone wants in a large, relatively pristine municipal park is another set of concrete steps in the creek bed, but rather have that step pool, natural looking, at least fishway, despite the fact that it's somewhat engineered, that provides passage, again, not just to salmonids, but other species as well. And of course, there's a myriad of additional assets and aspects to this restoration opportunity. There are educational opportunities. I think there's a K through 12 program that uses this specific project now as an educational opportunity. Place-based knowledge is heavily integrated into the project design and implementation. So the local tribe is heavily involved with this project as well. And yeah, with that, I'd like to acknowledge, of course, the Big Chico Creek Ecological Reserve, where most of this pre-restoration happens and where a lot of the post-restoration monitoring is going to happen. Chico State University, where both our BMI samples are analyzed, where our student volunteers come from, where we take classes out for field trips. And yeah, again, a lot of student volunteers, a lot of faculty volunteers that make this all possible, especially the earlier phases of pre-restoration monitoring. And then, of course, we got to acknowledge the funding agencies, first and foremost, the National Marine Fisheries Service, and NOAA, that made funding available for this restoration project, and CDFW as well, have been assisting with anything from native fish relocation and reintroduction to funding to permitting to field coordination and so on. And with that, hopefully we have time for a question or two. Excellent.
We have plenty of time. Thank you all. We appreciate seeing this team effort. Before we get into questions, I did want to properly share background information for Michael Helmar. He is a senior fisheries biologist at Fish Bio, and he is currently involved in the development of automated fish passage monitoring solutions and leveraging international partnerships to solve fisheries challenges. So let's give it up for Michael. And Madison Piori is a fisheries biologist, and she is currently involved in the pre-restoration monitoring efforts in Big Chico Creek as part of the Iron Canyon Fish Passage. So thank you so much for being here.
We can kick the questions off from the internets. Any chance you're measuring nutrient flush in stream post-fire?
We have not, no.
I see you back there.
Thank you. This is, I guess, kind of a follow-up to that question, but also related to the project as well. I think Holly alluded a little bit to keeping an eye on sediment transport, but I was curious if you are monitoring turbidity as well, especially post-fire, and how that may impact sedimentation in the project zone.
I am not a geomorphologist or an engineer. I have one. Yes.
Sorry, I didn't see it.
I will say that my engineer has promised that it's robust to sediment transport. Given that it's a stream simulation approach, I don't see how the project reach would stall out sediment any more than any other section of the creek above or below, given then that whole section is very rocky with salt rocks and sieves and all sorts of dynamic hydrology. So, do you want to add anything to that, Michael?
Sure. So that post-fire sediment runoff has been very dramatic at times, and it tends to be worse. It tends to be worst when we get a little bit of rain that is sufficient to wash sediment into the stream, but not enough to flush it out. So we've had, high turbidity concentrations. For that matter, for the most of this fall, this is actually the reason why for the first time we didn't do our fish survey this year. At first there was a health concern, there was an E. coli scare in the creek, and then we had high turbidity, and high turbidity continues to persist. It's really difficult to monitor. We tried initially to monitor some post-fire runoff, and the turbidity reading are off the chart. Like our equipment could not read the turbidity. If you're ever in Chico, I have a mason jar that I kind of use as a snow globe example with a water sample that I collected last Christmas, and you can shake it up and see what the creek looked like. Yeah, it was pretty dramatic, and that again just serves as an illustration, or just serves to highlight that we were positively surprised, even amazed, that both the BMI samples and the fish abundance survey last year post-fire after some turbidity events showed as high an abundance as we did see.
Could you tell us a little bit more about the non-salmonids that you're monitoring, particularly the freshwater mussels, the padofauna? What are you finding out there?
So we're working with Dr. Stuart Reed and the Xerxes Society to do mussel surveys. I can't remember the three off the top of my head. I definitely know it's the Western pearl shell. Do you know the other two?
The ridge and the floater.
Okay, the ridge and the floater. Stuart will be doing abundance and distribution surveys of those probably next year. And then post-restoration, he'll be doing it again. So TBD.
So, and maybe just to add to that, while the fish surveys have focused on fish, of course, in the past, the site selection for each year's survey is a random site selection within that ecological reserve. And what we've been doing throughout the years is noting observations beyond fish. So we can go back and look at where, in 2017 or in 2013, we saw mussel beds and what species they were, and we can go look for persistence at least. So we have a start.
We have plenty of time, so don't be shy.
We did get half an hour. Yeah, we did.
Okay, from online, the California Department of Water Resources manages a water quality monitoring station in Big Chico Creek, which collects data including turbidity.
Yes. Yes. They think they just reinstalled that or installed it. Yeah. Yeah, I used to not do turbidity until recently, though.
Thanks for that presentation. It's a really exciting area in particular. I think Big Chico Creek and Battle Creek, aren't they the largest strongholds for spring wren Chinook that are left in the state?
No, not Big Chico.
Not Big Chico.
It would be Mill, Deer, and Butte. Sorry, second. Yeah. Butte would be the largest stronghold, although that's largely due to a interbasin transfer from the West Branch Feather River. So a very altered hydrograph that benefits the number of salmon that come back to Butte Creek every year.
Any speculation on what the secret sauce is for spring wren Chinook in that area compared to other parts of the state?
Go for it, Michael.
So the unimpaired hydrograph is definitely has a lot to do with it. The access to higher elevation, deep cold holding habitat, basically. So anywhere else in these east side tributaries, all the larger ones, like thinking Lake Oroville, for example, block access to the higher elevation portions of the watershed, resulting in warmer water temperatures downstream and so on. So the big ones, Butte Creek has the largest population, but like Holly mentioned, it has somewhat of an, I guess you could call it an inflated hydrograph where water is transferred from the Feather River Basin into Butte Creek as part of a hydroelectric project. So that creates more habitat than there would be under natural condition, but be that as it may. The other two are Deer and Mill Creek. Those are two fairly, or at least were until somewhat recently, fairly robust spring wren population. And then Big Chico kind of falls right in the middle, right? So Big Chico Creek historically supported these fish, but then since the early 1900s when that rock fall happened, could have been an earthquake, there may have been other, could have just been natural erosion that blocked that access. That's when that consistency of that run was eliminated. So since fish couldn't access it or only access the upper watershed during a very narrow range of conditions. But now Big Chico Creek does receive these spring run salmon strays, whether they come, they missed a turn to Butte Creek and then take the next turn or take a turn early and don't go to Mill or Deer. We do see these salmon, and maybe I can go back a few slides here to that salmon hole slide. So this salmon hole here on the left, if you look very closely, there's probably 150 salmon holding in there. This was taken, I think, in May, if I'm not mistaken, and none of those fish made it through the summer. So right now, that's the best holding habitat they have available, but yeah, they can't go past that. And then you have all summer, college kids doing cannonballs and people snorkeling, and then the water gets really warm and they get stressed out. They don't eat all summer and they don't make it till spawning in the fall. But yes, to go back to your question, higher elevation, cold water is the secret sauce, and access to that, of course.
So I was just going to say, just having some experience sampling in that area, it seems like that area is really flashy. So you get these events like in fall or getting closer to fall where they just, a huge flash of scouring event or something. And the idea that, the fish and the bugs are doing okay. Maybe the sediment transport is quick and rapid, and then it's not really settling out. But yeah, it's just.
Yeah, during periods of big flow, those big flushes, it tends to clear fairly quickly after. It's that if we get that little bit of rain, that's just enough to mobilize some sediment or wash sediment into the stream, and then it doesn't flush out. It just lingers. That is, in my opinion, perhaps the biggest challenge to aquatic life. But yes, it's very, very flashy, very charismatic.
Before we release you all for lunch, one more comment from the chat. This person has burned off organics and suspended sediment samples post-fire, and the organics were 30 to 90% of suspended sediment. This may explain why BMI and fish don't respond negatively to post-fire. We will be starting back up promptly at 1:30. Like they're gonna start talking. So you gotta be in the room before that. So enjoy your lunch and we'll see you then.
Thank you.
All right, welcome back.
I'm still John Olson, and I'll be the moderator for this session. First up is Lindsay Metz. Lindsay is a lead scientist and program coordinator for the Community eDNA Monitoring Analysis Project and the Swamp eDNA Monitoring Programs. She's going to be talking to us today about eDNA and its future and in bioassessment.
Yes, awesome. Thank you so much. Hello, everyone. Very nice to see you all today. And also just a quick thank you to all the presenters from the first session. You guys were fantastic. So this is going to be a little bit of a switch up from some of the other presentations we've had this morning. I'm not going to be sharing any technical data with you all. Instead, we're going to be talking about future directions from more of a regulatory perspective, both at the State Water Board and other state agencies when it comes to our use of biological data and bioassessment data in our monitoring efforts and what that is going to look like, the integration of DNA and molecular-based tools. So I've got a few slides here at the beginning just to sort of set up the context. I'm going to go through these very briefly. But just to sort of establish all of the different ways that the State Water Board is currently utilizing biological data in our monitoring and regulatory capacities. Beyond just bioassessment, we are collecting a lot of different biological data types for many different reasons from the perspective of understanding the interaction between biology and water quality. And so there are many different ways that we're then utilizing that biological data in our actual regulatory processes. One of the big ones is going to be our integrated report, which is a biannual report that creates our 303D listing, which is the list of water bodies in the state that are impaired. And there are several different biological criteria that can be utilized to create a listing for a water body. There are also many permits that we issue that integrate biological criteria. We put out different types of advisories and signage related to biological information in terms of safety utilizing water bodies. And we also create a lot of different data visualization tools in order to share this information with the public so that people know how to safely interact with their water bodies. So just very quickly, we've already been talking about our bioassessment program, and many of you in this room are likely familiar with all of this information. But our current swamp bioassessment program is one of the cornerstone programs within our swamp unit here at the State Water Board. And right now, we are utilizing two different biological indices in order to evaluate stream condition. We have our CSCI, looking at benthic macroinvertebrates, and then our ASCII, which is looking at our algal communities. And then these two different indices are sort of what the basis, the foundation of both our perennial stream assessment program and our reference condition management program, which have been mentioned earlier today in previous talks. But as we've been going through the process recently of going through our program's sort of structure, especially in response to state budget related changes and the impacts of those budget changes, we've identified some different limitations and sort of considerations that we want to be thinking about and investigating as we're moving into sort of a new future for ourselves when it comes to how we're actually conducting biological monitoring here at the State Water Board. And some of the issues that we've identified in terms of our ability to have capacity and scaling within our bioassessment program includes issues related to the high cost of the laboratory analysis and the actual process of collecting field samples out in the field. Some of the procedures can be, is my mic coming in and out, or do I sound okay? Okay, cool. Sorry, it sounds weird in my ears. Some of the field protocols can be longer and more complicated, meaning that there's a more difficult translation to other external groups that we would want to work with in order to do biological monitoring. There can also be accessibility issues, particularly related to gaining permits to be able to collect live organisms in different locations. And there are some concerns in terms of the long-term availability of trained scientists to actually analyze the morphological data that we're generating. This has already become a big issue for us in terms of our algal community work that we've been doing. And we've also encountered, despite the fact that the CSCI and the ASCII have both been proven and shown time and time again to be robust indicators of stream condition, there have been some issues with actually translating that information when it comes to regulatory decision making. And we have received pushback in the past when it comes to actually implementing this data to create water body listings and to then therefore initiate mitigation strategies in various locations. And so that's something that we've been sort of fighting with and trying to figure out how can we start to create more confidence in using biology as an indicator of stream condition and actually create movement and management decisions related to that. So one of the new directions that we're exploring going into the future as an agency is how we can start to integrate new tools, in particular DNA and molecular-based monitoring strategies. So eDNA, this is a very broad definition, but this term has been used and thrown around a lot, I would say especially increasingly so in the past 5 to 10 years. And so just as a very general definition of what we're talking about when we're referring to eDNA, this is going to be any DNA that can be detected in the environment through water, soil, or air samples. So instead of collecting a sample directly from an organism, we're instead collecting an environmental sample and then analyzing the DNA in that sample that is naturally shed by organisms through many different processes. This could include things like skin cells, mucus, gametes during breeding seasons, waste, any kind of decaying material from live or dead organisms can all contribute DNA to the aquatic environment, and that can exist in many different forms, including live organisms if we're talking about microbes, DNA fragments. We can have DNA that's particle bound or dissolved in the environment, as well as whole intact dead or decaying cells. So all of these different sources and avenues for depositing DNA in the system can all contribute to the DNA we're finding when we're collecting and analyzing these samples. So just as a quick, very, very high level overview of what we actually do in order to analyze these samples. So we collect water from a water body, which has DNA contributed from many different organisms in it. And then we're going to amplify target DNA related to the organism group or groups that we are interested in evaluating. So for example, if we wanted to look specifically at fish, we can use primer sets designed in order to get the highest diversity of our fish population that we can hopefully capture within that sample. And for certain types of studies, that would be the end point of the laboratory process. So for example, if you're looking at endangered or invasive species, if you're doing any kind of fecal bacteria related work, microbial source tracking, if you're looking for one specific organism, that's where we're going to end our process, is by amplifying the DNA from that organism of interest, and then we can quantify the amount of DNA that was in that sample. If you're interested more in community level data, we can then go a step further and sequence the data in that sample. So we can amplify a region that is what we generally refer to as a barcode region for the taxonomic group of interest. And then we can compare all of the sequences from that sample to known sequences that exist in a DNA reference library, which then allows us to taxonomically identify all of those sequences in that sample. So then that's how we can generate community level data for different organism types of interest from a water sample. So just to give sort of an idea of one of the main advantages of working with DNA-based data, when we're using traditional methods, there's a variety of different techniques that would need to be implemented to actually look across multiple taxonomic groups simultaneously. So, if we're talking about microbes, we could be having to do culture experiments, particularly for bacteria. You could also be having to implement microscopy for either algal or benthic macroinvertebrate samples. If you're talking about larger plants and animals, you might have to start conducting visual surveys, doing transects and quadrat surveys, potentially, depending on the accessibility of the area. You might have to implement remote sensing technology in order to get in there to see what you can actually detect in that environment. And then if you're talking about even larger macro organisms that might like to run away from you, then you're looking into things like having to do electrofishing or trawling for fish, having to do deploy bird nets or bat nets if you're talking about mammals or birds. So there are all of these different technical strategies that we would have to implement to look across multiple different taxonomic groups within a single ecosystem which all require technically trained individual to implement. Whereas with eDNA, all we have to do in order to look at across these taxonomic groups is analyze a single sample for multiple different primer sets that would identify different taxonomic groups of interest. So we can collect water, soil, or air samples, and we can, from one single sample, look at fish and birds and bacteria, depending on what our interests are for that study, and really start to get at and understand community-level interactions and relationships in a whole new way that would be really difficult in using more traditional techniques. eDNA is also a really great complement to our traditional data. So it's not as to imply that we would be replacing these traditional techniques, as there's always going to be a need for boots on the ground visual identification of organisms. That's how we get really important information about the abundance of these organisms in our system and different life stage and health information. However, eDNA can be really effective in identifying organisms that would otherwise be discrete in the environment because of issues like cryptic taxa. Whereas in addition to that, traditional methods can also identify organisms that maybe haven't already had a reference sequence in a DNA library in order to identify those. So by employing these techniques in tandem, we can actually really increase the power of our ability to get a broader picture of the organisms within our system from more of a biodiversity community level perspective. So we've already been investigating the use of eDNA-based tools when it comes to bioassessment and biological monitoring. And we've been working very closely with SCWRP, particularly Dr. Susie Thoreau has been working with us in development of a molecular-based ASCII index. And the need for this really came about due to the lack of available algal taxonomists to be able to analyze these samples. And so that was an increasing concern. And we recognized that there was going to be a problem if we didn't come up with an alternative solution. And so we have, in tandem with SCWRP, we've been working on piloting some studies to determine whether or not an algal, molecular-based algal index would work out in the real world. So Susie's team has been piloting some different primer sets that are targeting different sections of the algal genome to determine their effectiveness, as well as looking at both taxonomic dependent indice options and taxon-free indices, and sort of testing their performance against our traditional algal index. And we've had some really promising results so far, and we're continuing to investigate that into the coming years. And just as a kind of a breakdown of the comparison between these two methods and why we might be interested in moving in the direction of a DNA-based index. So first of all, the cost, it's substantially cheaper in terms of the cost per sample to analyze an eDNA sample versus a traditional morphologic sample. And so that really means a lot in terms of our ability to scale up our efforts because we can collect more samples for a lower overall cost. There's also a lot more of the field effort in terms of collecting our traditional samples. It's a lot more arduous. It takes more time, more personnel, and has a lot more of a complex procedure and different types of equipment that might not be available to everybody in order to collect those samples. Whereas with eDNA, with just a syringe and a filter, 1 to 2 people can go out and collect water samples that can be analyzed for DNA data. The laboratory analysis component, there are increasingly a high number of labs that can process molecular samples, whereas we are increasingly losing taxonomists, and that's one of our big concerns going into the future. And in terms of the turnaround time of the data, it can take a long time due to just the effort that it actually takes to go in and do a microscopy analysis to get data back from that sampling effort, versus with DNA, it can take 1 to 2 months turnaround time for our data. So all of these things mean that the scalability of eDNA is really broad, which is really appealing to us as something that we can, oh no, I'm going over time, I'm sorry guys, that we can try to implement in the coming years because we're limited in our capacity to do more bioassessment monitoring than we are currently doing. So I'm going to try to breeze through these next slides. I'm so sorry. So the current applications of DNA at the water boards, we're already using it for FHAB pre-screening, looking for toxin genes in the environment. We have been collecting for the past 5 years community level biodiversity data with our community eDNA monitoring project. We are, especially our Region 9 office has been using eDNA to look for invasive and endangered species. And we've also been using microbial source tracking in tandem with traditional fecal indicator bacteria to look at fecal contamination mitigation across the state. So the two main potential future applications that we're going to be investigating in the next couple of years are the potential for a DNA-based benthic macroinvertebrate index alternative, not to replace the CSCI, but to complement the CSCI, as well as the potential for a DNA-based biodiversity index that would take a multi-taxon approach to look at stream condition and health. So with a DNA-based benthic macroinvertebrate index, it would have to be an entirely new index, so a new version of the CSCI where the foundation would be DNA data. And we would be largely building on the work that we've already done in establishing this molecular-based ASCII score. The main advantages are that it eliminates a lot of our future concerns around funding, access to taxonomists, our ability to integrate communities into our efforts, which increases our scalability across the state greatly. It's minimally invasive and requires no permits. You get data a lot faster, which means you can act on that data more quickly, and it's much more cost effective. But the main challenges are going to be that there are large gaps still in DNA reference libraries for benthic macroinvertebrates, and we need more work done on modeling the fate and transport of DNA in the system, which is basically how DNA moves in the water and how it decays over time. And we also need to establish more defined protocols for our field procedures and our laboratory procedures so that that can then be taken and applied statewide confidently to be able to integrate data from multiple different sources. And that is all work that is ongoing currently. There are several labs who are working on reference library gaps and fate and transport modeling. And in the next couple of years, our goal is to have well-defined field and laboratory procedures for DNA-based data. There's also this whole new concept, which is the DNA-based biodiversity index. This is something that we're really particularly interested in, as it allows us to look across multiple taxonomic groups simultaneously in order to evaluate the health of an ecosystem. And there's a really great example of this that's currently being implemented in New Zealand. There's a lab in New Zealand that generated this Tiki index. It's a stream health index that integrates, it's a taxon-free index, meaning that it does not actually taxonomically identify each sequence, but instead looks at the composition of the entire sample to determine how many sensitive and common species are in that sample and determine a stream health score from that information. And this has been implemented with such success that they've actually replaced largely their traditional bioassessment methods with this new Tiki index, and they're implementing it nationwide currently. And this is my last slide, so thank you guys for giving me a couple extra minutes to talk. But really, I just want to drive home the fact that the State Water Board, our regional boards, and several other state agencies have been investigating the use of DNA, and we really see this as a promising direction for the future of biological monitoring across the state. And so in the next few years, there's going to be a lot of movement in this area. And if anyone in this room is interested in this, has been doing work related to this, those are people we would love to work with and talk to so that hopefully we can, as a collective, learn how we're going to integrate this new technology into our systems. So thank you.
Thanks, Lindsay, for we're going to do one online question and one in the room question. So make it a good one and raise your hand real high in the room. Someone says it's a very promising approach for obtaining biological data. What are your thoughts on how to balance the increasing use of eDNA with funding challenges associated with its processing? And what potential strategies or solutions do you see for addressing these constraints?
So that's actually a really great question. And one of the things that both labs that we are currently working with, as well as, on like a national and even international effort, is determining sort of standards, minimum standards for eDNA data that can help us to start to drive down costs in terms of the materials needed to collect samples, the lab processing fees, by having these sort of minimum standards established. The lab that we are just started a new contract with, the Cal eDNA lab at UC Santa Cruz, one of their stated goals is that they really want to figure out how they can reduce costs substantially from where they are currently so that they can increase sample load, as much as possible. So this is both a goal on behalf of the labs who are doing the sample analysis, as well as folks like myself understanding how we can make this accessible to everyone.
We have an in-the-room question.
Wait for my mic. There's 80 fans on the internet that want to hear your thoughts.
I guess I'm old school, but I'd look, I'm just, the way we used to do, we get a taxonomic list, you get relative abundance. And so you make a very compelling case. I really like it. How does that compare? I mean, can you get relative abundance? Do you have to pick and choose the species, because you have a real limitation there? You might get a list of 90, normal taxonomy. So your limitation is the number of.
Right. So definitely. So part of it is, it depends on your application, because there are a lot of different applications for DNA-based data. I highlighted quite a few in terms of, if you're thinking about endangered species, if you're thinking about microbial source tracking, all of those different applications have a different need when it comes to having actual abundance values and the significance in that, it's the application of that data to make decisions. So when it comes to anything where you really need to have hard abundance values, it's always best to pair DNA-based tools with a traditional study. Oftentimes, DNA can be a really great pre-screening or preliminary step to identify the likely presence of an organism in an area so that you can concentrate your efforts where they're going to be the most meaningful. In addition to that, there are quite a few labs that are working on modeling the relationship between the number of sequences in a sample and the number of individuals in the environment. So that's something that we can hopefully estimate a lot more accurately in the very near future. But then also when it comes to some of these, when we're talking about like a biological index, especially from the standpoint of doing a taxonomic-free approach, at that point, we're getting a little bit further away from abundances and looking more at relationships. So from that standpoint, that we're thinking less about abundance overall. And so that will be less of a foundation of building an index like that than with something like the CSCI. So it really is just totally dependent on what direction you are ultimately trying to go with the data that you're collecting.
Thank you, Lindsay. Next, we have Sarah Stinson. Sarah's going to talk to us about using genetic approaches for studying mussels.
I'm short. Thank you for having me. I'm Sarah Stinson, and today I'm going to be presenting some research on behalf of myself, my colleague Aviva Fisk, and my supervisor Sarah Brown and Melinda Bearwald, related to our efforts to genetically monitor the golden mussel and the invasion in California using eDNA. I doubt I need to spend much time convincing many people in this room that the San Francisco Bay Delta estuary is an important habitat for many different species, including threatened and endangered species, specifically fish. We work with a lot of fish in my lab. That's why I put them on there, but obviously other taxonomic groups as well. Also, it's been highly impacted and altered by human activity, including factors such as climate change, poor water quality, and other stressors. So it's vulnerable in that sense. In another sense, it's vulnerable to invasives because of the physical connectivity of the system. And within the system, there's high physical connectivity, as well as the input of outside inputs like shipping lanes, for example. So there's the opportunity for invasion from outside ecosystems. So one of these invasives is the golden mussel, Limnoperna fortunae. And the reason we are concerned about this particular invasive is that it causes ecological and economic impacts to our system. I've listed some of the ecological impacts here. It is an ecosystem engineer. It disrupts the food web. It depletes phytoplankton and zooplankton. It can increase the likelihood of fish parasites. It changes the water chemistry and can also encourage cyanobacterial blooms. Excuse me. There are also economic impacts that are important in our system. So one estimate from 2022 was that the global cost for other invasions worldwide amounts to about 140 billion US dollars in damage. So we do have some economic impacts there in other systems. How does it affect these systems physically? It causes biofouling. It can impair water conveyance. There's physical structural damage that occurs. And as you can see on the left side of this slide, this is a flow station in the San Joaquin River near Buckley Cove. The crew that maintains and monitors this flow station noticed some inconsistencies with the data that indicated that it wasn't working properly. They went out to investigate, and lo and behold, it is just encrusted with mussels. So you can just get an idea of how physically they can just alter a substrate or structure that's important for monitoring or water conveyance. And then the timeline for this, I think it had been a little more than 6 months since they had checked that station previously. So this is happening rapidly in our system. The golden mussel is endemic to China, and the spread of its progress through various ecosystems is represented on this invasion timeline here. It really began to emerge as an important invasive species, especially in South America. These dates are approximate because we don't necessarily know exactly when they were first introduced, but we know when they were first detected and then recorded in the literature. So as the invasion spread through South America, that's where we're getting a lot of the documentation from some of the infrastructure damage, the economic damage that was occurring in the system. We do see a gap between when the organism was first detected in South America and then when it finally arrived in our system here in October of last year. However, it shouldn't be a surprise that it came here because of its success in invading other similar estuarine ecosystems. So to give you an idea of the invasion response timeline that we observed, so we don't know the exact date that the organism first entered our system. However, on October 17th, a field crew did notice that there was this unidentified species of mussel at one of their check stations in Stockton, near the Port of Stockton. They took photographs and samples. They sent those photographs and samples to various organizations, including the Smithsonian Environmental Research Center, the UC Davis Genomic Variation Lab, CDFA, and then the Department of Water Resources Genetic Monitoring Lab. And so there was this flurry of activity at the end of October where we were obviously trying to rapidly confirm that this was indeed the golden mussel. It's pretty visually distinct from what I've been told by taxonomists, but again, it's good to have the genetic confirmation. So this happened quickly. We got the genetic confirmation on the same day from both UC Davis and CDFA. And then that was consistent with the initial visual confirmation. So we knew what we were looking at was the golden mussel. So I work in the Department of Water Resources Genetic Monitoring Lab. So we began some initial qPCR testing shortly after the visual detection. And then obviously other groups, as they learned of the new invasive began their own monitoring programs. I don't have all the exact dates listed on the slide, but there are several other groups who are now also doing their own genetic monitoring with eDNA, including those on the screen here. So I think it's perfect that my talk was after Lindsay's, because I don't need to introduce you to qPCR or metabarcoding, or necessarily defend the importance of genetic monitoring as a complement to taxonomic identification. But I think everybody can agree that early detection is key, especially for managing such an aggressive invader as the golden mussel. So most of what we're currently doing now is using quantitative PCR to look at sites where we might be able to detect an early invasion. So we know they're in California, we know they're in the State Water Project, but we're continuing to monitor and get samples from other groups that are monitoring various areas of importance as far as water infrastructure or ecological importance to see when they're arriving, and that could maximize our efficiency in mounting a response in those areas. This involves a lot of collaboration and communication across different areas. So the various genetic monitoring tools that were involved to date have been taxonomic ID, so that's Sanger sequencing, looking at tissue-derived DNA, and then also eDNA monitoring, which I'll talk about a little bit more in a later slide. So the samples that we've been getting at our lab consist of townet samples, which you can see, this is a sampling event at Pyramid Lake. And that is similar to like a zooplankton net. We're getting this composite environmental sample. And as Lindsay mentioned, an environmental DNA sample can be various sources of DNA. We know that the immature life stage of the mussel is free floating, and a lot of our samples contain these veligers. So they are whole organisms in addition to potentially free floating DNA. And our laboratory protocols are pretty standard. We're using established kits for our DNA extraction, Kaya-amp for the townet samples, the blood and tissue kit for the single veliger samples. We're using the Pia et al. qPCR assay. And I added on this slide that this has been optimized with the help of the CDFW genetics lab. We've been through a lot of proficiency testing with the CDFW lab to really increase the sensitivity of the test. So initially, we got the assay, it was working, but we've spent a lot of time trying to maximize our effectiveness at being able to detect down to a single veliger. And a single veliger, if you've probably some of you have seen them, they're microscopic. They can be difficult to taxonomically identify. It's an early life stage. And so some of the samples we're getting are just one single veliger in a tube, and we can extract the DNA and then confirm if that is or is not a golden mussel. That's been very helpful with some of these samples. We also have done some specificity testing with this assay. So we wanted to make sure that the assay itself wasn't going to be presenting any false positives. It's not going to amplify with another mussel that's already present in our system. So we got a panel of different tissue extracted DNA samples of both invasive and endemic freshwater mussels known to exist in the San Francisco Bay Delta. It does not amplify with any of those. We were not concerned, but we did that just as a confirmation. And then once we process a sample, either a single veliger or a townet sample in our lab, we're also sharing those results with other organizations such as CDFW, who have compiled this really helpful map, also kind of scary map of the spread of golden mussels throughout California. So this map is available online. It's showing both the physical detections as well as the eDNA detections that are confirmed. And I zoomed in on the Stockton, San Francisco estuary location to show you that there's really extensive sampling that's been performed by us and other groups, and that we are getting a lot of detections in this area. And sadly, also now we're getting detections that have moved down through the State Water Project all the way to Southern California as well. So the mussels are here to stay. but we are monitoring them and trying to mitigate as best as possible. So I told you about our current monitoring efforts, but I want to talk a little bit about future monitoring efforts. So this is a great example of the utility of using genetic monitoring. And we should have expected they would have arrived at some point because of that lag that I showed you in the invasion timeline between when they were first detected in South America and when they finally made it to California. So it was really an incentive to be more proactive about invasive species monitoring. So DWR is starting a new project for surveillance of eDNA for invasives called Sentry. And we're hoping to incorporate both a targeted and an untargeted approach with this program. So what that means is we have a list of like our most wanted, right? We know some really bad invasive species that have yet to make it to this ecosystem or are just now beginning to be detected here. And we definitely want to make sure that our testing methods can target those species. However, we may have other organisms that we're not yet aware of that could cause ecological and economic harm. And so by using metabarcoding, a more untargeted approach, we can both potentially detect those other organisms that we're not yet aware of, and also go back to our archived samples and be able to go back in time and look at those samples or that data and see, were those organisms there when did they show up, basically, in the system. So that's a really powerful aspect of that type of genetic monitoring. There's also several different interagency groups that are working on this, and we're trying to maximize our effort and collaborate with those other groups. There's a golden mussel species identification interagency workgroup subgroup. There's an eDNA subgroup. There's the molecular methods workgroup that Susie Thoreau and Squirp facilitate. And so we're really interested in collaborating with other organizations to make sure that we are doing our best to keep track of the golden mussel and other invasives that might be entering the system. And with that, I listed some of those groups on this slide. However, I know there are others that aren't included, and I'd love to reach out and talk to anybody here who's interested in being part of that effort, or potentially who also has archived samples from the Port of Stockton prior to October 2024. I think it would be really interesting to be able to go back to those eDNA samples and see if we could actually detect them with our eDNA samples before they were physically detected. So talk to me about that if you're out there. And with that, thank you very much.
Do we have any questions for Sarah?
I have two questions, actually. So on your distribution map, I saw a point in Livermore, right?
Yeah, and
where is that coming from? Or
that's a great question. These data points are uploaded with lat-long data to CDFW's ArcGIS website. I don't think that that site is one of our sampling locations, so I can't answer that for sure. Yeah.
Probably New Zealand muds now.
Yeah, yeah.
And my second question is, what about Washington and Oregon? Have they been found up there yet?
Not that I'm aware of, although I wouldn't be surprised because of the speed that they've been moving through our system. Water temperature does play a factor in how rapidly the veligers are being able to be dispersed. So we've noticed there's some seasonal variation, like the mussels are producing veligers in warmer water conditions. So maybe they'll have a reprieve until next spring, maybe.
Before our next in-person question, someone on the internet, see, this is why we speak into the mic, people. They said the Livermore Point is maybe the South Bay Aqueduct?
That is possible, yeah. Okay.
Hi, great presentation. So I'm with the Bishop Paiute tribe. We're on the other side of the Eastern Sierra, and so the golden mussel, we're starting a task force just so because we've detected it coming up, and we get a lot of boating and stuff like that.
Yeah.
How early do you think we should be testing these waters for? And one of the reasons is we have a very intricate irrigation system that goes through the reservation, so we want to make sure that anything that's upstream in the reservoirs, it's not going to come down and eventually disrupt that whole system. So we're kind of how quickly or how early should we be, we do implement eDNA testing for the mussels, I guess
Yeah, I think earlier is better. I mean, we know that they're here in California, and so depending on the connectivity, like you said, there's a lot of boating that potentially could be transmitting those veligers from one site to another. They're microscopic, so you may not be able to see that there's anything physically on the boat. I know there's a lot of inspection points at various boating access points, but there's a chance it could get through. So I would say earlier is better. But yeah, maybe we could talk to you afterwards and connect about that.
Time for one last question.
Hi, thank you. You said that you have mitigation and rapid response when you detect these. Can you speak a little bit more into what that looks like once you detected them, and do you do anything about it?
Yeah, I knew someone was going to ask that. I am not directly involved with the mitigation efforts. There are other people within the Department of Water Resources and other agencies who are focusing on exploring the best mitigation approach. They're very difficult to eradicate. I know there's like a hot water technique where you can boil them away in certain water infrastructure locations. Obviously, that wouldn't work in the estuary where you have other species you don't want to boil. But I don't have the details on that, I'm sorry. But yeah, I can find out and let you know if you're interested.
Thanks, Sarah.
All right, thank you.
Next, we have Jonathan Juarez. Jonathan's just earned his master's from CSU Bakersfield, and will be talking to us about the effects of an invasive on leaf decomposition.
All right. So I'm going to talk to you guys about my thesis research today, talking about Atlanthus altisma, leaf decomposition in different freshwater ecosystems and its impact on aquatic macroinvertebrate communities. I wanted to start by acknowledging the land where the research was conducted is the traditional land of the Tohono Indian tribe. The Tohono Indian tribe is a consolidation of the descendants of various tribes in the area, including the Yokuts, the Kawiasu, and the Kitinemuk tribes. We're grateful for all the indigenous people who continue to care for and remain interconnected with these lands. Through research and conservation efforts, we promise to be conscientious stewards of these lands for future generations. So let's talk about food webs and how they're interconnected. We think of this as a common example that we all probably know about bears eating fish in the environment. Terrestrial animals rely on these aquatic food sources, but that works the other way around. Fish rely on macroinvertebrates falling into the river or whatever that might be. But leaf litter is also an important resource for aquatic ecosystems. When I see this picture, I see a bunch of potential energy waiting in that system. But we asked ourselves what might be utilizing this leaf litter. Macroinvertebrates utilize leaf litter as both food and habitat. They're important to both aquatic and terrestrial food webs. They specialize to fill different niches in their environment. We classify these as their functional feeding groups. Starting at the bottom left there, we have a shredder, collector, gatherer, scraper, grazer, and predator. So we see a lot of different niches that they fill in the aquatic environment. Macroinvertebrates can also be indicator species for stream pollution. So if we see a lot of the mayfly there, second from the left, we can summarize that there might be a pretty clean system that they're coming out of. Because they rely on leaf litter, that makes these riparian zones an important part in macroinvertebrates. And riparian or streamside zones serve as a buffer between terrestrial and aquatic ecosystems. They can reduce the nutrient pollution going into aquatic ecosystems so they can absorb those nutrients before they hit the water. And healthy riparian zones support increased biodiversity of aquatic biota. Now, riparian zones can be easily affected by invasive species. And just a definition of an invasive species here is a non-native organism to an ecosystem whose introduction causes or is likely to cause economic or environmental harm or harm to human health. There's some examples here of some aquatic invasive species that we probably have all dealt with in the field at this point. Water hyacinth, American bullfrog, or brown trout. So we talked about riparian zones. We talked about invasive species. And my research subject seems to overlap here. Specifically, I'm talking about Ailanthus altissima, also known as tree of heaven. Tree of heaven is native to China and North Vietnam. It was introduced in the US in the late 1700s. Specifically in California, it was introduced in the 1890s during the gold rush. Looking at the distribution of tree of heaven, we see that it's been introduced and established in all continents except for Antarctica. Looking at this map here, we see that it's actually spread through most of the US, including parts of Canada. So let's talk about some of these traits that make tree of heaven a pain to deal with. They can spread through clonal root shoots and can regrow after removal. So if you cut it down at the stump, it'll grow right back from that stump. It can grow up to 10 feet in a year. It can use allele chemicals, think plant chemical warfare, to suppress surrounding plant growth and deter herbivory. And in California, this is pretty important, it's really drought and industrial chemical tolerant. So what do we know about tree of heaven? A little recap here. Tree of heaven is fast-growing, drought-tolerant invasive plant. It uses allele chemicals to reduce native plant growth and deter herbivory, and it can establish in riparian zones. But this led me to, when considering all this, I realized this knowledge gap. Little research has been conducted into how tree of heaven can impact aquatic ecosystems. This led me to my research questions for my thesis. Does leaf litter from tree of heaven decompose at a different rate compared to native California species? And are these decomposition patterns consistent across different freshwater habitats? And does leaf litter from tree of heaven support a unique aquatic macroinvertebrate community compared to leaf litter from native plants? And is pattern consistent across freshwater habitats? So for my research, I had two research sites. The first was a pond located on the CSU Bakersfield campus. And the second was a fourth order perennial stream, El Paso Creek, located in Tejon Ranch in the southern Sierra Nevadas. So the species that I compared tree of heaven's decomposition to was the Fremont cottonwood and the London plane tree. These leaves break down at different rates in their environment. In the case of the cottonwood, they break down really quickly. In the case of the plane tree, they break down really slowly. So I had three treatments for my research here, one with only invasive leaves, one with a mix of both of the native leaves, and one with equal amounts of all three leaf types. So to accomplish this, I collected all the leaves when they senesced naturally into their environment. They were air dried in my garage for a couple weeks. After that, they were dried and placed into these plastic mesh netting to simulate leaf packs that you would see in the environment. Those were attached to ropes, and those ropes were deployed into the two test sites. They were then later collected at regular time intervals where the macroinvertebrates were separated from the leaf sample and they were identified down to the family level whenever possible. The leaves were combusted to determine their ash-free dry mass so we can measure that decomposition through time. So, and starting with the leaf decomposition here, on the y-axis, we have a natural logged percentage ash-free dry mass remaining. On the x-axis, we have time in days. So we see leaf pack decomposition rates were different among the different treatments. Looking at the left side here, we're starting with the pond. we see that the invasive treatment decomposed 3.7 times faster than the mixed treatment and 5.1 times faster than the native treatment. Looking at the stream here, we see that the invasive treatment decomposed 3.8 times faster than both the native and mixed treatments. And looking at just the invasive species here across those two habitats, invasive leaf packs decompose 3.6 times faster in the stream compared to the pond. So just a recap of my first question here, does the leaf litter from Tree of Heaven decompose at a different rate than the California species, and are these decomposition patterns consistent? Yes, absolutely. Leaf litter from Tree of Heaven decomposed at a faster rate than both the cottonwood and the plane tree leaves, and these patterns were consistent across both habitats. Just a discussion here of what we might be seeing. Tree of Heaven leaves are less tough than native leaves and have low lignin and high nitrogen to carbon ratios. Also, ambient conditions like temperature, flow, all of those can accelerate decomposition rates, which might be why we're seeing faster decomposition rates compared to the pond. And with those leaves being less tough, they might just break down a lot quicker. Potential implications here of replacement of riparian vegetation with Tree of Heaven may lead to changes in leaf litter composition, and changes in leaf litter compositions may impact aquatic macroinvertebrates that rely on diverse leaf litter compositions. So since we're talking about macroinvertebrates, let's move on to that macro data. So here I have a list of all of my sampling dates. For the macroinvertebrates, we're going to be looking at 2 dates in particular. My early date, which was 12 days in, and my late date, which was day 53 of sampling. So we're going to start with density here. We just have some box plots. Once again, I have the pond on the left, the stream on the right. Macroinvertebrate density was different between the leaf pack treatments and colonization dates. Here we have the late invasive treatment in the pond has a significantly greater density of macroinvertebrates compared to all other sampling dates. While not significant, we see in the early date, we see that same pattern with the highest density with the invasive leaves moving down to the mixed and then the native. Looking over at the stream site here, you might notice a no data sample there. Those leaves broke down almost immediately in that stream, so we just have a gap in the data there. But we still analyzed this the best we could. Macroinvertebrate density was different between leaf pack treatments in the early colonization date in the stream. We see that same pattern with a high density of macroinvertebrates in that invasive treatment. Looking at the mixed and native treatments across time, we don't see any significant difference in the densities there. We're going to move on to community dynamics. This is an NMDS figure. If you've never seen one of these figures before, don't be too concerned about the numbers on the axes. We're worried about what these points represent. Each point represents all the macroinvertebrates found in one leaf pack. And the closer that those points are to each other, the more similar those communities are. So looking at taxonomic community structure was different across habitat and time. Habitat we have in the red and orange, and the stream is in the 2 shades of blue. And those color differentiations are the different colonization dates. Communities were similar between leaf pack treatments. But now that we're looking at the community here, let's look a little closer as to who is actually there. So we're looking at the relative abundance of the taxa in these leaf packs. Overall in the pond and the stream, we see these patterns begin to emerge. Chironomids were present in much higher relative abundance in the stream compared to the pond. In the pond, we see a greater abundance of Oligochaeta. We also saw a greater relative abundance of Cladocera and Ostracota in the pond compared to the stream. Now that we've looked at this overall, we can break this down into those colonization dates across those 2 habitats. So Starting with the pond here, we see an increase of relative abundance of Cladocera and Oligochaeta across those 2 dates. And we see a decrease in Ostracoda across those 2 dates. Looking at the stream, we see a decrease in chironomids from early to late, and an increase in both Limnophilidae and Simolidae. So we talked about those functional feeding groups. We looked at the taxa, so we can look at the function of this, the taxa that are there. Looking at those functional feeding group community dynamics, I promise this isn't the same figure you just saw. It just all kind of bared out really nicely where we see the same exact patterns across both taxa and function. So macroinvertebrate functional feeding group community structure was different across habitat and time, but we didn't see a difference in those leaf pack treatments. Looking at the relative abundance of the functional feeding groups, we see shredders and collector filters are present in higher numbers in the pond compared to the stream. And we look at collector gatherers. Collector gatherers are present in higher abundances in the stream compared to the pond. As you can imagine, because we had mostly chironomids in that stream, they really made up all that functional group, the difference that you're seeing here. Moving on to splitting it up by the dates, just like we did with the taxa, we saw an increase in shredder presence from early to late in the pond, and a decrease in collector filter from early to late in the pond. Looking over at the stream, we saw an increase in shredder and collector filter abundance from early to late. And we saw a decrease in collector gatherers, right? Which makes sense. We saw a decrease in the chironomids that are making up most of this collector gatherer group here. Recap of my second question here. Does leaf litter from Tree of Heaven support a unique macroinvertebrate community compared to leaf litter from native plants? And is this pattern consistent across freshwater habitats? Partially. While taxa and functional feeding group communities were similar between leaf pack treatments, the relative abundance of taxa and functional feeding groups were unique between the treatments, time, and habitat. Patterns showed that invasive leaf pack treatments supported greater macroinvertebrate densities, even though those weren't always wrapped up in the stats. We saw those patterns, which was really nice to see. Just a recap of the discussion on the macroinvertebrates. Tree of Heaven leaf litter may be an easy to access energy resource for aquatic macroinvertebrates, which may explain the high density of those macroinvertebrates in those samples. And homogenization of Tree of Heaven leaf litter may favor faster developing macroinvertebrates that can quickly exploit those resources, potentially leaving those slower developing macroinvertebrates hungry. Thinking about the mitigation efforts that we use on Tree of Heaven, Tree of Heaven's ecological impacts may extend past terrestrial ecosystems, potentially altering freshwater ecosystem processes. And we can think about comparing aquatic macroinvertebrate communities between invaded and non-invaded riparian forests to provide insight on how much of an impact Tree of Heaven may be having on that aquatic system. I just want to take a second to acknowledge all of the undergraduate support that helped me throughout this project. It was a lot of field work and a lot of lab work, so thank you to all of them. Thank you to all the members of my committee, including my advisor, Dr. McNish, who presented earlier, our collaborators at Tejon Ranch Conservancy. Special thanks to all my funding resources and Ms. Gail Beatty, who made a generous donation to my research, and the Tejon Ranch Company for allowing me to access the ranch to conduct the research there. And I think my time for questions is now. Thank you.
Any questions for Jonathan?
Hi, Jonathan. Neat study. That was neat.
Thank you.
Do you happen to know anything? I despise Tree of Heaven and the way it smells. Do you happen to have any knowledge about the allelopathic compounds that are involved? Like what type of chemistry that is?
I don't have anything exact, but I do know that a lot of those compounds are water soluble. So I think what ends up happening is when those leaves hit the water, it's almost like tea, right? Like it just ends up diffusing out of the plant. And there wasn't anything that bared out in the results that show that it affected the macro community or anything like that.
In the pond and the stream that you conducted your study in, was there tree of heaven present?
There's no tree of heaven present at either of those sites. Those sites were chosen mainly because they were easy for me to get access to. When you're a grad student trying to get done on time, you kind of take what you can get. But yeah, there's no tree of heaven anywhere near those two sites. Historically on the ranch, there was tree of heaven that they'd eradicated, but it's no longer there. And on campus, there is some tree of heaven, but it's not near the pond site.
I think it would be interesting to continue this and do it in a site where tree of heaven is established and see if you've got that difference in BMI community where there's those faster BMIs compared to slower life BMIs. And
yeah, and I think that, going back to that last slide I had there, where being able to compare between an invaded and non-invaded forest might provide a lot of insight on that, whether those communities have changed because of the presence of tree of heaven there compared to an uninvaded one, for sure.
Any other questions? Thanks, Jonathan.
Thank you.
So the next thing that's coming up is the poster session. So poster session from 2:30 to 4 this afternoon outside in the lobby. Come by and talk to our students and see some of the work they're doing. After that, we're also having our California chapter meeting here. So if you're interested in or you're a member of the chapter, please come by and talk to us for a little bit. And then we're having a mixer, which will be held at Locked Barrel, starting at 5 o'clock for early career and then 6 o'clock for the rest of us. But thank you all for attending this session, and I'll see you at the poster session.